3rd National Seminar on Nanomedicine and Medical Devices in Healthcare, organized by APJ Satya University, School of Pharmaceutical Sciences, sponsored by DST-SERB, Government of India in collaboration with Walter Bushnell, ASG Biochem, and Martin and Harris Global.
A famous saying goes like, when darkness comes, let us not condemn the dark, but light a lamp to illuminate. For that, I request all our Vice Chancellors, Professor Rajesh Dhankar, Dean Dr. Anupama Diwan, ma'am, and guests to come forward to lamp light.
Now I would like to invite Dr. Dean Anupama Diwan for welcome entrance.
May I request Professor Roopkar, Professor Klaes Hilbenser, Dr. Majumdar, and Professor Sanjay Ayuwal ji to please come on the stage. Very good morning. What a great day. I'm really honored today that we all have together here to celebrate the 3rd National Conference on Nanomedicine and Nanotechnology in Healthcare. I'm really honored to have a wonderful Chancellor ma'am who has been a source of motivation and guidance for all of us, for giving us always the encouragement to hold on the conferences, seminars, and do something different from the need. We are also honored to have with us Dr. R.S. Thankar, our Vice Chancellor, sir, who has been a constant encouragement for us to work in various aspects, not only the curricular, but also the co-curricular activities from time to time. Further, to excel ourselves, we have with us Pro Vice Chancellor Ma'am, Dr. Sucharita, who have been is so instrumental. She herself is an example to be followed in means of constant working at university. Pharmacy profession, you know, all is a very complex and well developed, requires a well developed set of competencies if you really want to have a rewarding career in life. So competency, do you think that it will come only from the curricular things which we have in hand? No, we really have to master the skills taught in the theory and practical examination. Not only that, but we also have been outrightly able to present ourselves do some research and present ourselves in the form of the seminar and conferences from time to time. We at SPS believe in holistic development, therefore, for the students and to integrate with continuous development of education and research activities of School of Pharmaceutical Sciences. We are today here to organize the National Seminar, which has been sponsored by DST-SERB on Nanomedicine and Nanotechnology in healthcare. It's the third initiative in the list which we have. We had two similar conferences in the past, which were totally offline, but due to COVID, we are having it today in a hybrid mode. Looking at the seminar schedule, having many wonderful and accomplished speakers, I am certain that it will be a remarkable weekend of learning, innovation, and networking. Some of the topic touched upon will be new and emerging opportunities in the medical devices, including biomedical sector, their vigilance, pharmacist role, responsibility. We are also touching upon nanocrystals, novel spray drying technology, and many new technology for which the stalwarts from the field, you name them, you will find, like Professor Alok Ray to begin with, who will be giving you insight on the emerging opportunities in medical devices, followed by Dr. Kalai Silvan sir, who will be talking on material vigilance program of Government of India, which is a new initiative by Government of India. We also have Professor Bansidhar. We also have many dignitaries from the industry throughout the day. We have 4 sessions, scientific session today, and 8 speakers will be there, and each session will be having the 2 speakers. During the lunch time, the posters which we have received will be displayed online as well as offline at campus, which will be evaluated by the 2 stalwarts from different universities who are participating. All of us are ambassadors and role models. Our decision, achievements, and guidance have the ability to influence our next generation. So, with my last words, I want to say, dear all delegates, please ignite your passions and ignite passion in others as well. I promise you, the reward and the impact will be transformative. Very good evening, very good morning, and once again, I welcome you all. Thank you very much.
Thank you so much, ma'am.
I would now invite our Vice Chancellor, Sir, Professor Rajesh Dhankar, who is currently working as Vice Chancellor at APJ Satya University, His professor, Dhankar has held responsible administrative positions like CEO, Higher Education, APJ Education Society, New Delhi, Vice Chancellor, Maharishi Dayanand University, Rohtak, Haryana, Vice Chancellor, Ansal University, Gurugram, Vice Chancellor, Amity University, Raipur, Director Center for Canadian Studies, Professor in charge, Faculty of Management Studies, South Campus, Principal, Maharaja Agrasen College, Saramati Postgraduate Men's Hostel, South Campus, and Chairman, Finance Area, Faculty of Management Studies, University of Delhi. In the past, he holds PhD and PhDs in the area of finance. Professor Dhankar, a thought leader and institution builder, is the ex-dean of the prestigious Faculty of Management. University of Delhi. Professor Dhankar has been visiting professor at Lakehead University, Canada, chair professor of finance at Ansel University, and is the professor emeritus, School of Management Sciences, APJ Satya University. Dr. Dhankar has been actively involved in teaching, research, training, and consultancy in the field of finance since 1977.
He also had been the recipient of Commonwealth Scholarship for PhD program in UK and postdoctoral studies scholarship for USA by the Government of India. He also had PDs in finance from John Anderson Graduate School of Management, University of California, Los Angeles, USA in 1987 and had taught in various foreign universities including University of California, Los Angeles. University of Southern California, USA, and Lakehead University, Canada. He has completed several major research projects with financial assistance from national and international agencies. 16 candidates have done PhD under his supervision, and a few are presently pursuing their work. He had 6 finance books to his credit and has published 125 critical research papers in the area of finance in major national and international journals. Dr. Dhankar is on the governing bodies counseling of various educational institutions and director and trustee on the boards of several public and private sector organizations. He is a member of various committees in the central and state governments. Professor Dhankar is on editorial board of several leading journals in the field of finance and management. He has been honored with several awards, including Best Vice Chancellor of the Year in 2016 and Haryana Ratna in recognition of his contribution to the welfare of society and Best Vice Chancellor. Over the stage, I would like to call him over the stage to motivate the students with his wise words and encouraging them.
Let me at the outset welcome all wonderful speakers and delegates in today's seminar. Also, I would like to congratulate our faculty of pharmaceutical sciences and of course led by Professor Anupma Diwan, a leader in pharmaceutical industry, for organizing this very timely conference. We all know a good healthcare system is very critical for the good health of any country and eventually the economy. And India is no exception. In fact, we need it rather very badly. We saw the importance of this during the last 2 years in the COVID-19 crisis. India is a leader in pharma sector, and we have proved it by producing very timely and very early 2 COVID-19 vaccines. When we think of healthcare in any country for that matter, 2 things are very, very critical. Availability of medicines at affordable price and a good infrastructure where medical devices plays a very, very important role. I'm not a student by any stretch of imagination of pharmaceutical area, as a few points been made about my background. I'm a student of management. But we all have in our lives seen and realized the importance of healthcare. In a poor country, particularly, if the medicines are not available in time and at affordable price, you pay a huge price for it. Therefore, we have to really ensure that there is a fair play and strong regulatory mechanism in this area. Equally important, as I said earlier, is the healthcare devices. We visit hospitals and somehow or the other, we all end up saying, I wish we have better infrastructure, particularly government-funded hospitals and medical centers. I think this is something that we really have to, as an expert in this field and policymakers, have to really somehow find a way out. And I have a sense that like IT industry, from early on, we focused on software, not the hardware. And we became leader in software industry. People know us all over the world. Our students and graduates have gone all over the world in leading multinational IT industry and developing the best of the softwares. Because we took a lead into that and ignored the hardware part of the IT industry. And therefore, we have lagged behind. Of course, of late, we have done some good work, but in hardware from early on, we had to import. I don't know, I do feel that somehow the same story holds in case of pharma sector as well. We are importing most of the medical equipments and which are costly, which are not so affordable. Hence, A, they are costly, and B, as a culture in our country, we have a poor maintenance systems. Therefore, the medical devices that we end up having, A, are not adequate, B, are properly not maintained. And it is in here, to me, this today's seminar on nanomedicines and medical devices in healthcare is very relevant. And I'm sure the speakers would really conferences are important, but to my mind, if we can have our goals in two ways. One is help educate the students and practitioners about this importance of these two things: the availability, affordability of medicines and maintenance of good medical devices. And somewhere the great leaders that we have today in the conference, of course, and I welcome them again, Professor Alok Ray from AIIMS and Professor Roop Khar. I'm sure these eminent people are in all policy-making bodies, and we have to really articulate our thought process and see to it the government policies be at the department level or at some regulatory bodies like PCI and others. We articulate the importance of it. Only then we would be able to take care of the health levels and needs of a large country like India. Having said that, but to me, equally important is also to give importance not to that much. Of course, modern medicines is very important, and somewhere we feel that we can't do without it. Like we realized during COVID-19 crisis, how these two vaccines that we developed and some other countries developed, how they have saved millions and millions of lives around the world. But I think we should also take pride and try and preserve and follow and develop further our alternative systems of medicines that we in our country from ages we have had, be it Ayurveda or Unani or some other very famous alternative medicine systems from the south of our country, that from so many years that we have found that our ancestors have made good use of it and have really saved hundreds and millions of lives. And also it brings to me the thought that I want to share is, let's not get sick at the first place. Why have so much, why we have to have the need of so costly medicines and equipments? If we can live a life which is healthy and we don't get sick. So wellness, health wellness, literally has to be properly given the due importance, and infrastructure and should be made part of the course curriculum, to my mind, that how to live a healthy life, how not to get sick. That's where the whole wellness concept becomes relevant. So we need to really see to it that at the first place, we prevent illness by following a proper health wellness system across the country, right down to the families, right down to the schools and universities. And then, of course, we should be also ready and sort of the infrastructure and medicines and other support system in our medical centers and hospitals should also be available in time at affordable price. These are the thoughts that come to my mind today on this wonderful occasion when our School of Pharmaceutical Sciences have organized this one-day national seminar on nanomedicines and medical device in healthcare. I once again congratulate Professor Nupma Diwan and our faculty for organizing it, and welcome from the bottom of my heart, on the behalf of APJ Satya University, on my personal behalf, all the eminent speakers led by Alok Ray from AIMS, New Delhi. With these words, I inaugurate this one-day conference and wish a great success. Thank you so very much.
Thank you so much, sir, for your wise and encouraging words.
Now let's move on to the scientific session which we are here for, which is the third national seminar on nanomedicine and medical devices in healthcare with our great speakers. Now I would like to invite our first session chair, Dr. Roop K. Khar, Director, BS, Anandpuriya Institute of Pharmacy, Faridabad, Haryana, India. Professor Roop K. Khar is renowned academician and a research scientist. He served in various administrative and academic positions for more than 35 years at Jamia Hamdard in the capacities of Dean and HOD of Faculty of Pharmacy, Dean Student Welfare, Placement Officer and Protector of Jamia Hamdard. He has supervised 80 PhD, 200 M.Pharm theses. He has published more than 300 research papers in international and national journals, 11 textbooks, and is the inventor of two US and 15 Indian patents. Presently, Professor Khat is Professor and Director of B.S. Anandpuriya Group of Institutions, Faridabad, NCR. He is Director of leading pharma manufacturing company, Unicure India Limited, and Executive Director of consultancy company, Formulators Concept Limited.
Now, Now I would like to invite Dean Dr. Anupama Diwan ma'am to welcome the chair with a green certificate. So our first speaker, Dr. Alok R. Rai, was professor and head of Center for Biomedical Engineering, Indian Institute of Technology, Delhi. He was also concurrently professor at All India Institute of Medical Sciences, New Delhi. He was Forgatory Fellow of National Institute of Health, USA, at Center of Polymeric Implants, University of Utah, USA. He was a fellow at University of Liverpool, UK. He, he was a visiting professor at All Union Cardiology Institute, Moscow, University of Leiden, Netherlands, and Max Planck Institute for Polymer Science. Now,
I would like to invite Dr. Dean Anupama Devan ma'am to welcome the speaker with the green certificate.
So you are most welcome, and since you are on online mode, so we will be sending it through YouTube. Thank you very much, sir.
Our first speaker topic is new emerging opportunities in the medical devices and biomaterial sector in India. So you there?
Shall I start?
yes, sir.
Yes, sir.
Very good morning, Honorable Vice Chancellor, Sir, Professor Dhankar, Professor Anupama, Dean Ma'am, Dr. Majumdar, and distinguished faculty, and my dear student. I want to share the screen now.
Now you can share your screen, sir. You can share the screen, sir.
Can you see the screen?
So we cannot see the screen.
Hello.
Sir, you can share from the green button there, this share screen.
What happened? I can't. Can I share my screen?
Yes,
Yes,
sir,
sir,
there
you
is a green button at the bottom, share screen.
Okay, just
Click
hold
on
on.
that, open the PPT, and then you can share.
Just hold on. There is something wrong. I just can't share my screen. Can you see my screen?
Yes, sir.
Yes, sir.
Okay. Now it is okay? Hello?
Yes, sir.
Can you,
Yes,
can you
sir.
see my screen?
Yes, sir. We can see it.
Okay. This is the area. I will just start with the summary. The economic growth of a country mainly depends on its capacity for technological innovation. The technological advancement not only require generation of new knowledge, but also how this acquired knowledge can be converted into marketable product. What is in our language, Arthakari Vidya. That knowledge which will bring money. India's track record in this endeavor has been insignificant. Besides this, the Indian medical technology industry is still a nascent compared to other Indian industrial sector as compared to the medical technology industry in USA and Europe. The given favorable demographics and the rapid expanding technology base and increasingly venture capital flow to the India, it is anticipated that the major medtech industry will develop in India over next few years. If suitable ecosystem is created, the main requirement of this ecosystem are the trained manpower and new concept in the area of medical devices. Currently, almost all devices are imported. At present, we are manufacturing majority best material used for these devices, but we are not producing any of these medical devices. One of the major lacuna in is the trained manpower in this sector. USA and European countries have well-developed academic program in this area. This type of program requires people from the several disciplines to work together. This interdisciplinary program will equip students of the diverse discipline equally well take up the innovation and entrepreneurship role in the private industry as well as academia. The vision is to make India a manufacturing hub for affordable medical device by 2025. This is the summary. Let's start with the main thing. As you know, if you become richer, you will live long. Something like that. Life expectancy in India is around now 70. It was few years back, 62, 63. And China is better than us now in reaching around something like 80s. The moment you can afford better medical facility, your life expectancy will increase. I will tell you some of the things now. If you look at other animals in this world, most of them lose their capacity to live or will die the moment their capacity to reproduce has gone. For a human being, this is around 45 years. After 45 years, so-called in a general term, the warranty for your life is over. After 45, you may have a heart problem, you may have a problem with your eyes, problem with your kidney, with several part of the system, because your capacity to reproduce has gone according to the nature. But we people like to live long, and there is no end to it, as much as we want to live. So to make this living, say like worth and painless, you have to replace most of your parts. Something like that. You can replace from your teeth to your heart, to your kidney, almost all part of your thing you can replace. So take the simple system like your heart. If you want to make an artificial heart, this is the one of the things is that at the first early of my life, when I was in America, I started with the first artificial heart, making artificial heart at Salt Lake City hospital. So this artificial heart required materials, advanced wireless system, biology, mechanical engineering, and software. People from all these area together can make the heart, an artificial heart. Yeah. Actually, look at the small thing like what is you have a heart attack, something like the heart is a pump which gives blood to all your system, including itself. They usually this is gone through the something called coronary arteries, and because of your food habit, age, this coronary arteries are clogged. So therefore, you have to require a replacement, which is usually the bypass. Or now what we use a stent, which can open up your coronary artery. These are the stents. These are essentially metallic spring, either coated with polymers with the drug or uncoated. It is just kept inside your artery so that it will be open up and the blood can flow easily. Okay, you can also replace most of your dental implants, say like all of you. Actually, I cannot look at you. This is one of the problems in the webinar. And you can replace all your dental system. You can lower limb, if you lose for any reason, you can also replace with artificial limbs, with your hand, upper limbs. So look at the world pharmaceutical industry. It is something like 800 billion. And medical device is around 220 billion US dollar. But India, it is a very, very small amount. Only 15% is the only medical device. But if you look at, we have 1.3 billion people, out of which only 250 million people can afford medical device or a dental implant or a heart surgery. This is 25 crores people. Even that, they require a large number of materials. Say like, look at intraocular lenses. Something like that, the figure you read, how big is the figure? Contact lens, retinal surgery. We require large number, vascular graft, heart valves, pacemaker, blood bag. Our annual requirement is very large. And hip joints, knee, shoulder joints. You see the number we use for a year. Catheters, oxygenator, and you now know because of the COVID how these are now very useful. I will just take one simple device, which is when you have a problem with kidney, we use kidney dialyzer. It's a very simple instrument which only filtering system with a pump. Although all materials which are used in this instrument, something like polysulfone, polyacrylonitrile, polyamide, all we make in this country, but we don't make this instrument. But now the effort has been made to make available in this country. Almost all polymers which are used in medical devices and diagnostics, something like Dacron, Lycra, Teflon, nylon, elastics, PVC, polystyrene, polyurethane, etc. They're all we make in this country. There is no problem in that. But so something like the biodegradable polymers, you pharmaceutical student must be knowing that slow release materials are made from this slow release of drugs, or you have to coat this stent with the biodegradable polymer. These are the polylactic, polyglycolic acid, they are copolymers, and all other things are we produce in the country, but we never produce the final product in this. Because it happens because people from pharmaceutical, from hardware, from microelectronics, microfluidics, from people from nano device do not work together. This requires a joint work. Single individual cannot produce medical devices. So what actually, something like around 2007, we take this challenge. At that time, no idea of Atmanirbhar Bharat. We think that our students were successful in making software engineers, but nobody attempts to become a medical device product or the startup. So in 2007, I, with along Professor Balram and other faculty members, started a program which I was leading to make medical device available in this country at affordable price. So what do? We take the challenge. Usually research are two types. One type of research is that whatever you like to enjoy doing something and you have the interest, you do it. And mainly to produce very high quality paper in the high impact journal. But this requires different type of challenge. We decide, if you decide to make a medical device, any device, you have to first generate idea how this device can be made very easily. Then we gather people, expert in this area. Then we make some research. Then we test our idea. If it is successful, we will go public. If it fails, then we use the same cycle. So this program started. It's called the Indian Biodesign Program. It was supported. It was.
वो लेके आओ तुम अपना एक्सट्रैक्ट थोड़ा सा लाना.
Is there a problem?
No, sir, no, sir.
Collaboration partner was Stanford University. And what we do, actually, the program started 2007, 2008. We take people from the different background. So like I will only 2008 I have seen, this is continuing. People from, say like Amit was an industrial engineer, Jayant was a product designer, Nitish Chasmawala was a plastic engineer, Sandeep, Professor Sandeep Singh, presently is as a cardiologist in AIMS, and say Jaggu was an electronic engineer. We get a group of people. Like that, several years, we have people of different background, you can see them. And the first year, we have the theme was emergency medicine. Then theme was pediatric care in 2010. Then gastro, GI tracts, like this. What we do, we take this 4 or 5 students. What is it called? Is called clinical immersion. We send them just to see around how the surgery is done, how the patient is admitted. Just don't ask any question, just observe. After observation, we send them to community medicine center where we have several places. So they go and just observe how the medicine is dispensed, what is the difficulty. And then we ask, what you can do? You'll be surprised to know, in the 2008, they have come with something like 456 idea. They told that we can make new 456 new instrument to ease of surgery and other things. Then we do. So we thought that 456 is a very large number, which can be sold in the market, a feasible outcome in the market. Then we screen, it comes to be 172. Then we next did, we called the physician and asked these young entrepreneurs to present their idea and told them, if you make this instrument, are you will be interested? 50 instrument was approved by them. It was again, it is too much. So we thought that only 4 or 5 students, we only thought that we will take the 15 and we'll, among them, we'll see that which can be better idea. So I will only give an example, few example. Today only 3 examples because lack of the time. Sometimes when there will be physical, I will show you all this thing. So one of the emergency need was there. When a patient arrives, if you cannot see their vein or their arteries, how do you introduce liquid in the emergency patient? Usually it is given in the bone. So there was no instrument available for doing this. So the start that how easily you can introduce liquid to a bone by the doctor. So experiment started. As you know, all engineers, they wanted to different way to make a hole. They started with the wood. Then the idea was given that if you take an egg, if you can make a hole without breaking it, I hope that will be the best. So you can see this now. This is the final one of the final product. So finally, this is the final product. I will also choose all the other ideas so the student can have. Other idea was like that, it failed. The another idea with another example was that with the multiple pin. So all this failed, but the one which successful were patented. With the multiple pins. Sometimes it worked, sometimes it didn't work. So once this instrument was successful, we did it cadaver. This is a clinical experiment done with the cadaver. So this is the one which we patented, and it will be now available in the market. Next one I will show you. Before going to this, I will give an idea. The way you cannot control your urine, some of the patient cannot control their stool. So there is nothing available in the market to handle if you cannot control your stool. So therefore, an instrument was developed, and this is a very successful one. And this instrument now available not only in India, it is available both in Europe and in America. These people have become very rich. They are not selling there because they think that company is a very high value company now. And we will go to the idea is that there are large number of patients which cannot control their stool. Something you can give, they can either their nurse can their help, or they can do that with themselves. I will see the instrument is something look like this. I will show the video, that will be much easier to explain. And this is something in a plastic device which can go through your anus, and then you can deattach it. And this can be taken out. Then this can be attached to a, okay, this is taken out. This is something like, and can be, and this is something can be collapsible. So therefore, liquid stool, air, and solid stool can be collected in the bag. And the bag, you can go to a toilet, bathroom, and you can separate out. Usually it will collect 1 kilo. So therefore, nobody can know that you have a problem, that you cannot control your stool. This is the different type of component which are made to this. This is like a umbrella, reverse umbrella. You can, and the one you saw that he took out, these are the parts. And this is now available in the market. Indian market is like this, and the product in US and the European market, because they have a problem with the plastics, they should be degradable, all these things. The price is quite high compared to Indian market. This is the one is available, and this technology was transferred to this these 4 guys who made this thing. And okay, this is the consumer, the company. You can find out in the website. If you have a in your home, you require this, you can purchase from this. In their website, you will be cheaper than the market. The next one is a very simple device. Usually, if that accident happened in the road, there was to immobilize the lower limbs. The things were not available in a lower cost to the public. So 3 of our students were interested, who started their own company now. This is something like that. If there is accident happens, it's very difficult to set there in the place. So they develop a very simple device. This is made up of disposable splinter, made up of say like the package, say like your fridge comes to your home with the package, something like that, cardboard material. And this is very cheap and available, and you can use for the left or right, anywhere. And this is now the technology is licensed to our Hindustan latex, and they have given that name Rigo, and is available. It is look like in the market like this. almost Karnataka, Maharashtra, and some other South Indian states, in their emergency vehicle, it is available because it's cheap, and you can take home with the patient, and it can also be used in the X-ray, and it has not to be changed. Okay. The next another device I will tell is a very simple device. They have developed a one type of bed sheet which very easily you can transfer patient from one bed to other bed. It was very difficult earlier, and this is also technology has been transferred to a MGM company. And okay, the another thing which has taken international international attention, when the baby is born, you have till it will not cry. People will, in our village, will think that it's not alive. So therefore they take it and start. What happened? Usually a cough is in their neck area. You will understand it. I will show the usually it is done like that. If the no electricity, that are suction pump helps. So therefore we develop a handheld pump which can easily be used in our villages. Then the system was later improved, and then it was scale up. So now the instrument is available in a handle, and which can be used by your pedal. And this was have a lot of international, because it was very cheap, available can be operated by the ASA worker. So it have attention. So the Hillary Clinton called these students and appreciated their work when they were exhibiting in America. And then many other things we have also developed. I will not go in details, just will tell that when child is born, it's very difficult to know he is capable of hearing or not. They have developed a small instrument which ASA worker can use. This is the instrument, and can know if the child is hard of hearing or not. This is the instrument which is available now. And that is a small torch is available for the to look at the arteries and veins of the kids, a newborn. This is the torch. And okay, these are several other devices are available. You can go to say like Sanford India Biodesign Program, or search in my name. You can find out all the instrument we have developed. The lack of time, I am not telling anymore, because the 40 minutes is the time. So what has happened now? Our capabilities have begun to catch up with our ambition. So the future is going to be much better than the present. I have the students, when I was playing cricket like this, and now things is happening like this. So the great change has been occurred in this country, and I hope. Thank you for your attention. Please open for any question you want to ask.
Now I want to draw the attention of Dr. Rupkekar and Dr. Alok Arde to have a brief discussion.
Dr. Alok Ray.
Yes, yes, sir.
Thank you very much for an excellent presentation on a very, very contemporary topic which concerns our country quite a lot now at this stage. Government of India has also arisen, and we are paying quite a lot of attention. Development of new technologies in medical devices, new parts, API and as well as medical device industry. Now you have very nicely highlighted the importance of technology and innovation, and you have said that our scientists, pharmaceutical scientists and other scientists should understand any technology which they develop, it should be possible for us to get it translated, something which is useful to the society. That is very, very important message I want to convey to my teachers and my technology scientist friends. Sir, you have given very good examples of various illustrative examples of how medical device industry is happening in all over the world. It is dominated in USA and European countries. Even now we are importing most of these devices from them, and we need to do quite a lot of hard work and quite a lot of efforts. It needs a push from the government and also the practice-oriented approach from the scientists. You have given some very, very good examples, sir, of coronary artery bypass graft, of dental implants, of applications in ophthalmology, of intraocular devices, of contact lenses, cardiovascular devices, dialyzers, ultrafiltration techniques, synthetic polymers and biodegradable polymers. I can highlight here for the scientists. This area is going to be very good for pharmaceutical scientists to explore. There's a lot of effort, possibility of developing devices with the help of biodegradable polymers. And then another important thing, Dr. Ray has said that it's important to be able to have medical devices at affordable prices. And that is the call of the Indian scientists, because I think, if we are not able to do it, a common man, it's not going to be accessible to a common man who cannot afford it, And you have very rightly said your carry home message, and we are very highly impressed, sir, by your video illustrations. There were some very, very good examples. They'll definitely provoke the thoughts of various different scientists and be able to develop some technologies and assist and convert their science, convert their thought processes into something which is useful for all of us. And your take-home message is very clear, that you say that our capabilities have begun and we are beginning to understand what we have to do, and certainly there's going to be a very, very good future. So I just wanted to say these few words so that our young scientists, our young teachers, professors would understand. And I have a data that they say, you see, what is the value of pharmaceutical industry at the moment, By 2025, as you said correctly, the medical device industry is going to surpass that value. That is the scope of this industry in next 10 to 15 years. With these words, I again, sir, thank you very much. So nice to see you, and your lecture was very thought-provoking. And I think it will certainly spark thoughts of many of our scientists. And if there are any questions and any thoughts and anything you want to ask Professor Ray, here is an excellent opportunity for all of us to interact with him in the online mode. All of you, either online or offline. कोई भी कुछ सवाल अगर किसी का है, कोई कुछ पूछना चाहते हैं, तो प्लीज यू आर वेलकम.
आप हिंदी में भी पूछ सकते हैं.
My question, sir, to you would be, can you hear me, sir?
Sir, yes. Mike.
My question to you would be, sir, I am Dean of School of Journalism and Mass Communication. So I would like to know that we all live in India as a poor country. We have been independent almost for 70 years. I just want to know, why haven't we been able to do this what we are doing now earlier? Why we make medical devices affordable to the poor of the country up till now?
I can't hear clearly. Can you just repeat it?
Sir, Professor Ray, he wants to understand why in last more than 5 to 6 decades, we have not been able to pay our attention to medical devices in the sense that it is affordable to a common man. Am I right, sir? Right. Yeah.
Sir, even other industry, say like even pharmaceutical, we only are coming out from this because we are not capable, because our industry people will not invest money in research. It is only academician doing research, and academician have a very limited time to do it. We were not prepared for this. We are just coming out of this because we have slightly become richer. We can now afford. I can give an example. When I joined as a lecturer, I never dreamt that I will have a car. I only dreamed that I will have a scooter. Now, now you know what is the way. Medical industry is a very top industry. It's quite difficult to produce a medical device. It's much easy to produce a software, much easier, because it requires a lot of people, infrastructure, which we do not have.
तो उसको हाँ तो हमने कोई भाई थोड़ी ना है, उससे हमें पता चल जाएगा एटलीस्ट कि वो कितना उसी से
that is why we have not been able to achieve what we had. But it's happening now.
There is some underlying reason, Professor, I will tell you. I can compliment, I will tell him based on the data. You see, pharma industry, medical devices now are classed as drugs. Medical devices are drugs as per Drugs and Cosmetic Act. So pharma industry has grown over a period of time, last 5 to 6 decades. You know, there's very serious problem even with the pharma industry, that active pharmaceutical ingredients, we are heavily dependent on imports from China and other countries. So you see, our pharma industry is going to fall down after 10 years if we don't pay attention. So Government of India recently has paid attention to two things. One, most important, is to develop the API industry. It is related to medical devices. That is why there are already identified as 4 or 5 parks for API. And similarly, 4 or 5 parks for medical devices. You know, these parks mean that Government of India is going to pay for the central facilities of such parks, you know, all those water, I mean, electricity and everything, so that the cost of that API or a medical device is going to go down.
This is like special economic zone.
Exactly. That is what is happening. And I think, you know, already so many medical devices are available, which are Indian companies, which are available at very, very less price, including those cardiac stents and everything. That's what I wanted to give you, that clarification. Thanks. Thank you, sir. Thank you. If there's no more questions, no, I think we again thank you, sir, very much. Thank you for your time, and thank you for your excellent presentation and contemporary, bird's eye view of this very important topic. Thank you very much, sir. Thank you.
Thank you, sir. Thank you so much.
Now I would like to invite Dean Dr. Anupama Diwan to felicitate sir with Certificate of Excellence.
Foremost, sir, we are really sincerely thankful to you for accepting our request for being over here. As rightly said by Professor Khar also. Sir, your words have been a really great source of encouragement for all the budding pharmacists. We would love to have you over here offline.
Thank you so much.
When we get sober, you please take out some time to visit our university. For the time being, online, we are again submitting the honor to you, sir. Thank you very much.
Thank you. Thank you so much for a kind invitation.
Now I would request all the participants over here who want to register on the spot can go to the admission department to register.
Our second speaker, Dr. Kalai Selvan. He is the Senior Principal Scientific Officer, Indian Pharmacopoeia, Ministry of Health and Family Welfare, Government of India, Sector 23, Rajnagar, Ghaziabad. He received his undergraduate and postgraduate degree in pharmacy from the Tamil Nadu Dr. MGR Medical University, Chennai, and PhD from University of Delhi. He has 22 years of multitude experience in the pharmaceutical sector, such as academic, drugs, standard setting, clinical research, and medical devices. Currently, as Senior Principal Scientific Officer at IPC to protect and promote quality standards. He works towards establishment of medical product safety, surveillance system such as pharmacovigilance and material-vigilance program of India. Now, I would like to invite Dean Dr. Anupama Dewan ma'am to welcome the speaker with green certificate.
Now, our second speaker topic is Material Vigilance Program of India, Roles and Responsibility of Pharmacy Profession.
Respected Chair, Professor Kar sir, respected Anupama Dewan ma'am, Dr. Majumdar, other faculty members, esteemed colleagues, and dear students. Very good morning to all of you. It's my great pleasure to be with you. And I'm very happy that the APJ would able to arrange this workshop during the current COVID pandemic by following all COVID measure protocols. It's a great honor for me also to be here and to speak something related to pharmacovigilance or vigilance for medical devices safety surveillance system. So this is what I thought of sharing with you. And it's a great initiative taken by APJ Group for selecting this topic, nanomedicine and medical devices. I'm sure nanomedicine related things will be discussed tomorrow, but today the medical device is very encouraging one. You might have listened the first speaker, craze, how the medical devices are being designed and how it is being marketed. And you might understand now how about job opportunities, career opportunities for all of us in the sector. As chairperson also, Dr. Rupkar sir also mentioned, there are a number of medical devices industries are coming out in India. There are a lot of units are being set. You might be aware that in Delhi NCR, Jewar Airport is coming up. They are also a special economic zone. Medical devices. Other than that, we have number of medical device industries now, like Andhra Medical Technology Zone in Visakhapatnam. It's coming up like anything. Currently, we are fully dependent on import only. The medical devices are imported, And gradually over the period of time, maybe 5 years or 10 years down the line, I'm sure the medical devices are also being manufactured in India. So this is how the background about the medical device sector is concerned. But now what I am going to discuss with you, there are good initiative by the Government of India, Ministry of Health and Family Welfare, to how we are going to ensure the safety of medical devices, whether all the medical devices are safe, or do we are anticipating some kind of adverse events with the use of medical. Produced in the market in the year 1960. That time, the phocomelia, which is one of the severe birth defect, was not known to the community or to the entire healthcare professionals. The phocomelia, should I continue? Yeah. Yeah. It's okay. Yeah. So the thalidomide was best known example to start pharmacovigilance, and the pharmacovigilance started in 1960 itself. After the thalidomide tragedy, the WHO started an international program for pharmacovigilance, where now currently all the member countries, more than 150 countries are participating in the international program for drug monitoring, including India. So that is how the pharmacovigilance system is being established for drugs are concerned, drugs or medicines or vaccines are concerned, after thalidomide tragedy. But what about medical devices? Are we anticipating, are we are expecting the medical devices are safe? No, because this is another example you can see. There are number of adverse events, serious adverse events, even revision of surgeries. So this happened because of the hip implants. One of the leading manufacturers product was introduced in the market in the 2009 and 10. And the year end 2010 and 11, it was found that the hip implants were causing serious adverse events because of metal leaching and other related issues. The quality of metals or materials used in the designing or manufacturing of medical devices plays an important role. And you can see the devices, the particular company devices were recalled from the market because of the revision of surgery, because of the inflammation, because of the death, etc. So this was the case study. One of the case study was shown to all of us that yes, there is a need, there is a vigilance even for monitoring the safety of medical devices also. Because here the pattern of adverse drug reaction reporting or pattern of occurrence of adverse event are entirely different from the device, drugs. As we said, the medical devices are manufactured or designed by using the metals or ceramics, as Sir mentioned, biodegradable polymers, and other materials are used in the devices. You can imagine when the metal quality is not proper, there is a chance for metal leaching. Most of the medical devices like orthopedic implants, knee implants, hip implants are manufactured by using metals like cobalt, nickel, cadmium, etc. But we must ensure it's like a API here. Here, the quality of the metals or materials used in the devices should be ISO standards or Indian Pharmacopoeia standards or Bureau of Indian standards. When the quality is not met, then definitely there might be a chance of adverse events. So you can imagine now how the medical devices safety is important. So this is one of the example I would like to mention here. Like this, number of examples are available, case study are available. So therefore, like drugs, there is a need for pharmacovigilance or vigilance for medical devices as well. So that is the reason we have a system now. Currently, the Government of India, under the Ministry of Health and Family Welfare, we have a stable and governance structure for both pharmacovigilance and medical vigilance. Here, I would like to mention why a stable and sustainable program, because government patronage is there, government commitment is there. Both the programs are supported by the Government of India, both in terms of administrative support, financial support. We have a separate budget for these two programs in the health budget for pharmacovigilance and material vigilance to run this program. That shows the commitment from the government level. And to run this program, we have a funding now. Yes, we have a stability. We have a resources for this program. And who is going to run the program now? The question comes, a national center, a dedicated national coordinating center for these two programs. So on the lines, as per the recommendation of WHO, we have established a national coordinating center for both Pharmacovigilance Program of India and Material Vigilance Program of India, that is at Indian Pharmacopoeia Commission, where we are coordinating these two programs. And then support from the National Licensing Authority, who is the regulator. Here there are two things. Indian Pharmacopoeia Commission is a national coordinating center which provides scientific based data, which evaluates the data, comes from the clinical setting or comes from the industry. Those adverse events are analyzed, assessed properly, and now communicated to the regulating authority, enforcement agency, who is the CDSCO, that is Central Drug Standard Control Organization, is responsible for taking any action based on our own data. Here, the best thing and the milestone achievement which I would like to share with you here. Currently, the regulatory authority, that is CDSCO, is taking action based on our own data. Maybe let it be a drug-related action or a device-related adverse drug reaction data, decision making. You might have seen that box warning, label change. So all this happens based on our own data, taking our own data, India-specific data. It was not happened before 2010. Our regulator, our CDSCO, took action based on the data available either from US or Europe or any other country, which may not be necessary. Scientifically, there are a number of reasons. The pattern of ADR occurrence may differ. There are a lot of pharmacogenomics factor, food habits, many things. So we have to have our own data. So now it is happening because of these two data. And we have a legislation, we have a separate rule for medical devices, that is medical devices rule, we will call it as MDR 2017. It is being introduced in the country, first time in the country. Now all medical devices are regulated like drugs. All medical devices are approved as drugs. Now let us come to, this is actually general definition of pharmacovigilance for as per WHO statement. We as a professional, as a pharmacist or as a doctor or as a nurse, so our responsibility is, as a pharmacovigilance professional or as a healthcare professional, our responsibility is to monitor the safety of not only drugs, vaccines, medical devices, and even traditional system of medicine also. That is also need of the hour to monitor. So this is about the general vigilance system is concerned. But again, this is the rule actually. Medical devices rule was introduced in 2017. and it is effective from January January 2018. So here all the devices are regulated now by the CDSCO, Central Drug Standard Control Organization. The regulatory part is concerned. Regulation means you know very well, right? Approval, marketing approval, export, import, manufacturing. So these are all the part of the regulation. And now the safety also comes under the monitoring program, monitoring the safety of medical devices, right? Now, how we are going to classify those devices? Now we know there is a regulation for medical devices. All medical devices are approved as drugs under the Medical Devices Rule 2017. Actually, there are 4 classifications. All the medical devices which are used for the disease diagnosis, treatment, prevention are classified under this 4 category. That is called Class A medical devices, which is also known as low risk category. Then we have a Class B, low to moderate risk, then moderate to high risk, and then high end medical devices, high risk medical devices. So here Class AB medical devices are like absorbent cotton, sutures, ligatures, needles. So all those devices are low risk or Class A or B category of medical devices. Then we have moderate risk of medical devices like intraocular lenses, cataract lenses, which are classified as a moderate to high risk. Then if you talk about Class B or high risk medical devices, all implants like cardiac stents, orthopedic implants, knee implants. So all such devices are put under the category of Class D devices. Other than this, the diagnostic kits which are used. Now, of course, each and every one of us might come across the COVID diagnostic kits. I think we all are familiar with the diagnostic kits. The diagnostic kits which are used in the COVID, like kits, thermometer, pulse oximeter. Many devices are also considered as a drugs or approved as a drugs. These all are diagnostic, in vitro diagnostics. So this is about the overall risk-based classification of medical devices currently as per the Medical Devices Rule 2017. Now, how we are going to collect the data actually? What is the system do we have to collect the data? I mean, adverse drug reaction data. We have established centers where a hospital or clinical setting, that is called regional monitoring center. It may be a hospital or it may be a institution affiliated with hospitals where we have at least pharmacology, pharmacy or biomedical engineering department is there. Then those institutions or those hospital can function as a regional center. So national level, sitting at national level or capital, you can't do anything without having the data from the field. We need to have data from the field or hospital. So therefore, under this program, Material Vigilance, we have established currently 150 regional centers along with 500 plus centers of pharmacovigilance. So all these centers are working in collecting the adverse drug reaction or adverse event data with the use of medicine or medical devices. So these data will be analyzed here. This is another area where we all need to acquire a lot of knowledge and continuous training. How we are going to analyze the data. Even one nurse or one pharmacist can report anything. Report an adverse event is one thing. That is the culture of reporting. Once it is reported, we as a center, we as a National Pharmacovigilance Center or Material Vigilance Center, our role is to assess the report, that is causality assessment, temporal relationship between the drug or device and the event. We need to assess that. Whether the adverse event is because of the drug or because of the medical devices, or sometimes the adverse event may be disease pattern. Progression of the disease also may induce some adverse event. So that is not related to device or drug. That as we as a pharmacist or we as a core team, we should have a knowledge and adequate skills to rule out or to conclude this event is because of the device. So that is called the causality assessment. So all these things are happening under the program. So then you need to communicate to the authority for appropriate action. If it is really this adverse event is related to drug or device, then you take a call. Otherwise, just keep on monitoring. So this is how the program of actually the program is functioning at Indian Pharmacopoeia Commission. Again, this is a system which I would like to already have mentioned the system to you. Now let me come to another area how we are going to work together for collecting the adverse event with respect to our drugs or devices are concerned. Like we need to work with the clinical experts. If an adverse event related to ocular devices, then naturally ophthalmologist on board. We need to discuss whether we are working at the regional level or national level. Clinical experts or specialists are very important when a medical device device category is concerned. Then biomedical engineers or clinical engineers. Say for example, if an adverse event with the use of pacemaker, then we as a pharmacist, we cannot take any action because we have limited knowledge about pacemaker or ventilator, etc. So there you need to discuss with the clinical engineers or biomedical engineers. That is how the biomedical engineers are very important stakeholders or group when we do plan any research or when we plan any study related to medical devices safety is concerned. Of course, pharmacists will be there. Then hospital manager or management, lab technicians, patients. So all will be there in the study or the medical device safety is concerned. Then reporting part is concerned. The question comes whether we should report only serious event or non-serious event. Everything to be reported. Seriousness criteria, as per the WHO, we have different criteria for seriousness, maybe death, life-threatening, prolonged hospitalization, congenital anomalies, etc. And non-serious, all remaining adverse events are considered to be non-serious. So the serious and non-serious adverse events are common even for medical devices, vaccine or drugs are concerned. So the reporting is concerned, both serious and non-serious to be reported. And while making a study, when you want to plan a study for medical devices or drugs related adverse event reporting, quite adequate volume of information needs to be collected in the reporting form, adverse event reporting form. It may be patient related detail or drug or medical device related detail, manufacturing detail, and then description of the event. So all these things are required to be captured in the reporting form or online reporting system is concerned. Then only we can take a decision or we can analyze the report, how it is being correlated each other. So these are the basic and minimum information required for a system is concerned. Maybe a regional center or national center, minimum information needs to be captured to this form. Actually, this is according to the global adverse event reporting of medical devices concerned. And there are some differences when we collate, analyze or collect the adverse events. There are some differences between drugs and devices actually, even from the point of clinical trial onwards. Very well. So this is a quite simple way I'm trying to just differentiate how both devices and drugs are different in terms of adverse events reporting part is concerned. This is a very brief analysis what we did. So related to the medical device adverse event reporting, the pattern of adverse event assessment and analysis goes this way. Procedural error, maximum adverse events are related to the procedural error. Maybe this requires to be continuous training and hand-holding support to the healthcare professionals. It's a preventable one and avoidable one also. Procedural error may be a breakage, device expulsion. So these are the common things which we could able to conclude at this point of time. So it's very early to comment. This is just 2-3 years old program, and we are just getting the reports, and this is a trend analysis. And I'm sure over the period of time, we may have different kind of factors which influences the adverse events associated with the medical devices actually. And what about the roles and responsibilities of healthcare professionals in monitoring these adverse events? Who can do what? Whether biomedical engineers only alone can do, or pharmacists can do, or combinedly do. So this is kind of our based on our experiences. We have just attempted how pharmacists or doctors or nurses can play in this role, you know. So we have some limited knowledge or limited information in our curriculum, pharmacy curriculum. We might not complete, you know, medical design. Some basic knowledge based on our curriculum and our practice, we can able to manage the adverse events with the use of drug-eluting stents. We can manage hip implants, ocular lenses related adverse events also can be able to manage and report it by the pharmacists and other healthcare professionals. But when it comes to other instruments or equipments like ventilator, in vitro diagnostics, X-ray machine, MRI, scan machine. So there, I think biomedical engineers can only play an important role in monitoring and reporting of these adverse events. But naturally, yes, it may be a collective responsibility, and we can able to manage also. Actually, for the industries, we have introduced this field safety corrective action form. If any medical devices is found faulty or not performing, then it is the responsibility of the manufacturer or industry to provide the batch number, particular medical device and batch number in the form of field safety corrective action form, so that the remaining samples available can be withdrawn from the market. So this is how the field safety corrective action form is being developed, and it is also being introduced in the CDSA website also. And overall, the safety monitoring system for medical devices, the challenges are concerned. Reporting part is the first challenge. Culture of reporting. Still we are under reporting. Still where the drugs are concerned, still we are under reporting. By comparing the population, we have 1.3 billion population, but if you look at the reports, still we are under reporting. But if you take me medical devices, still it is low. The culture of reporting needs to be enhanced. To enhance the culture of reporting, adequate training programs, I think programs like this, awareness, then practice, we have to inculcate the culture into our practice. Reporting. Then introducing advanced tools like mobile app, online reporting, that is also being done. Mobile app we have introduced for reporting. And second challenge, database. We don't have specific national database for medical devices. The data are managed in the Vigiflow, which is the software for drugs. And currently the government of India is also developing the national database for medical devices also. So that becomes a tool for the regional WHO CRO's region to use this software for data analysis of medical devices. Then globally, we don't have, even global database also not available for medical devices reporting. For drugs, yes, we have a robust software for called Vigiflow and Vigibase to analyze the data. But for devices, we don't have a global database also. And last part, decision making, whether the data information provided by the doctor or nurses is sufficient to take a regulatory decision on medical devices. That is quality of information and evidence-based information. So this becomes an important challenge in the decision making. So overall, now these are the challenges, and we have to address these challenges also now. It's the responsibility of every healthcare professional and every pharmacist how we are going to address these challenges, particularly with the use of medical devices safety is concerned. Enhancing the reporting and generating the quality information or quality data, and developing a database for medical devices. Then only it becomes easy for us to analyze the data, so that we will have some baseline value, and we will it will be easy for us to take a decision. And last is decision based on our own data. Prompt decision making also should be put in place. I think thanks a lot. This is what my presentation actually over to the chair. Thanks.
I think yes. Well, that again was a very, very important lecture, very useful lecture by Dr. Vivekanandan Kalai Selvan in the offline mode. Kitta farak hai, offline and online, we are together. I think we are going to have these sessions more often after a month or so. मैं ये कहना चाहूँगा that you see, you all know that safety and efficacy are the primary requirements of any medicine or a medical device, and this safety of medical devices is classified under material vigilance. So it is so important, Now material vigilance has become also equally important as compared to pharmacovigilance. I think, our IPC is doing an excellent job. Officers like Kalai Selvan and many others. This is a main central pharmacovigilance center of Government of India, and he has given us as to what are different. Only one act which is there, MDR 2017. I think our students should perhaps go through the act and see what it deals with and what are the various, parts of this act. Then secondly, what he has highlighted, what are the various challenges in, it's very, very important. We all should be a part and parcel of pharmacovigilance and material vigilance. We should understand, you see, when any drug is given for a long time, chronic administration, there are certainly going to be certain adverse effects which are not reported in our country. हमारे यहाँ तो पहले data ही नहीं था. We were all importing all the pharmacovigilance data or using the pharmacovigilance data which was available from other countries, particularly US and Europe. But with the pharmacovigilance activity starting at IPC, we have made a very big step in this direction, and similarly material vigilance should also be having the same importance. And I would like to say to my students that pay attention to when you are reporting, be one of those officers who can report. You have to be an important observer. Observation is very important. हमारे घर में भी पेशेंट होते नहीं, बहुत से लोगों में डिवाइसेस भी होते हैं. तो कभी-कभी जब भी आपको, whenever you feel there is some effect which is classified under those 4 categories which Sir has said, that we should try to understand it. Is it worth reporting? And I would tell you that please do it, whether it is relating to the drug or whether it is relating to a medical device. Dr. Kalai Selvam, very, very wonderful. Thank you for giving us this information, and to the students and the teachers of this institution and all those who are attending this. In the meantime, if there are some questions, some doubts regarding the safety, regarding anything else as to how it is handled at one of the very important Government of India centers, IPC, he will be ready to reply. Yes, there is. Please take the mic, introduce yourself, and
Yeah.
Good afternoon, everyone. Sir, my name is Sharma, and I work with the Department of India. And the town which I want to ask is basically questions raised by the students. And whenever we do the IPC, so there is a PTA program, which is IPC is relevant. That question. So toll-free number as well as the website of PBPI. And can you find all the tabs or spirits of any pharmacist can register their complaints? What is the drop down which we extract while complaint about any medicine or about any
Great question. It's a good question. Basically, the reporting, the reporting is the suspected ADR reporting form. It is available in the website. By using the suspected ADR reporting form, the report should go to the regional ADR monitoring center, not directly to IPC. The channel of reporting, if you look at, I mentioned 500 plus ADR monitoring center. There are institutions, those who are having hospital, clinical settings, identified as a regional center. So basically, your report should go to the regional center. And the regional center will evaluate, will do the causality assessment. Then the report will enter into the VG flow. It will come to the IPC. So directly IPC reporting to IPC may not be possible. First, the report should go to the regional center. Say, for example, here, you have to see the regional ADR monitoring centers in this region. There are many centers in the region, like Apollo Hospital, Medanta. So you have to submit this report to the regional center. This is one thing. If you use mobile app, you can always options of reporting this report to any of the regional center. The regional center will see the data assessment, then the data will be entered into VG flow. There are some questions in the chat box. Yeah, let me see. No.
Thank you, sir. I'd like to invite Dr. D. Anwar to receive the certificate of this.
Maybe they are making others.
Now I would like to invite our second chair session, Professor Harvinder Kohli, Professor and Director, School of Pharmaceutical Sciences, Delhi, Pharmaceutical University, DST, New Delhi. She has been listed among the top 20 business development professionals out of 20,000 internationally who follows best partnering practices in recent European Business Developer magazine, November 2009, while at RAN magazine. She has close number of major licensing deals for pharmaceutical products and technologies, and also facilitated launch of more than 40 pharmaceutical drugs, including NCE, biosimilar, and technological barrier product to market for India and other emerging markets for RAN vaccine. She has excellent track record of creating diverse team structures involving research collaboration, in licensing opportunities across the all therapeutic areas.
Now I would like to invite Dean Dr. Anupama Dewan ma'am to welcome the chair with a green certificate.
Thank you for we'll planting a plant in Africa. Thank you, ma'am.
Thank you. Thank you, Dr. Anupama Dewan.
Now we would like to invite our co-chair, Dr. Majumdar sir, to come over the stage and receive this green certificate from Anupama.
जाना हो गया 12 बज गए चलो
Thank you. Thank you, Dr. Anupama. I think, first of all, I would like to congratulate APJ Satya to organize this webinar, which is on both online and offline mode, and a very important topic of a nanotechnology and medical devices, which is being covered in this. And this plenary sessions, we have keynote speaker, Dr. Manish Dewan, who is the head of the strategy partnership and entrepreneurships development of the BIREC, Department of Biotechnology, Government of India. And he would be talking on the growing startup ecosystems and the allied opportunities. Basically, when we are talking about the nanoscience and nanotechnology, both are very fast growing areas of science and technology. In the the entire spectrum of science and technology, whenever these advances has been taken up, there is need that the policy makers and the knowledge generation bodies or the knowledge transfer bodies or the application bodies work hand in hand. So very importantly, the policymakers, especially in the nanotechnology area, is the the various funding agencies like DST, DRDO, CSIR, DBT, BIRAC, DIT, ICAR or ICMR. And also when we talk about that, there is a knowledge generation bodies like R&D university or the research institutes. And there is a need for the knowledge transfer bodies and also, the application bodies of the nanotechnology areas. And definitely, the government has worked in this vision and also made, a lot of center of excellence, and BIREC is doing an excellent work, in this area. And this nanotechnology is the frontier technology worldwide. So this technology is, deals with not the nanoscale materials and the system device developments, but it requires, really sophisticated experimental fabrication, production, and testing facility. And the government of India has floated, the millions of rupees to establish these centers, and also, through the nanotechnology and through different funding agencies. So there is a lot of database which has been available, but definitely, this being a very interdisciplinary area, so it requires, amalgamation of the scientists, academia, and also the industries to achieve the objective. So in overall, the objective is not achieved, if we all not work. And this is the platform, where everyone is being together and discussing and finding it out that, what and how we can do together. And the establishment of the pilot plans for the, production materials. So in the previous, speakers, touched upon the medical devices and also the pharmacovigilance. And definitely, when there is a more nanostructured materials, it also requires, more comprehensive studies. So not the for doing all this, there is also a technology, science and technology mission throughout the country which take up these research challenges. But for everything, you need kind of a funding. So I think, I'm very great that, Dr. Manish Diwan is there, and, he is a known person in this area. He has worked himself, in a science and technology, and definitely now, spearheading the VIREC as a head. So I think over to Dr. Manish. I think prior to that, you would be reading his brief profile. Yes. Okay. Great. Go ahead.
Now we would like to have our third speaker, Dr. Manish Diwan, M.Pharm PhD Head Strategic Partnership and Entrepreneurship, Development and Make in India Facilitation, Cell for Biotech Sector, Biotechnology Industry Research Assistant Council. He is leading large national programs for Government of India's effort to nurture and empower the biotech innovation. Mission, especially Startup India, Make in India, contributes to policy reforms, strategic collaboration including international partnerships, serves on national committees and advisory boards, leads national flagship schemes and program of BIREC promoting startups.
Entrepreneurs and industry. He has about 27 years of global R&D experience in pharma biotech sector, from discovery research to product development, science administration and project management for industry and academia. Before moving into public sector in 2017, he contributed to several clinical drug candidates for global drug development at the Ranbaxy, Tower and University of Alberta, Canada. Also developed technologies during doctoral and postdoctoral research that were transferred to industry.
Now I would like to invite Dean Dr. Anupama Diwan ma'am to welcome the speaker in certificate.
Really honored to have you here, sir. So we will be planting a tree in your name, and just a token of gesture, we are extending our warm wishes to you. Thank you, sir, for joining us.
I think that's very nice of you. Greatly touched by this gesture. Thank you, Dr. Anupama. Thank you, Dr. Popley. I could see Professor Khar, Professor Majumdar, Professor Bansal, Professor Kohli, and greetings to all the fellow colleagues, panelists, and faculty and students. This is, as Dr. Popley just said, a very relevant and important aspect, deep tech technology development and knowledge generation. Knowledge which gets generated unless it gets translated and deployed for tangible outcome, reaching to the society, addressing unmet needs, it stays within the knowledge generation institutes, colleges, universities, or the centers. So there needs to be a concerted effort, an enabling effort, I think, involving all the stakeholders, as many of you are already there. So I represent an enabling body, which is Government of India's undertaking that enables and supports this ecosystem where the deep tech technologies can be nurtured and taken forward. In next half an hour or so, I will try to use a few slides and share with you what are the different opportunities and how the ecosystem is evolving, so that you in turn can can utilize that information and use it at your discretion to reach out to these opportunities. Just let me try and share my screen from here. If someone can prompt me that my screen is visible or not.
Dr. Diwan, it's visible.
All right. Thank you, Dr. Popli. So I will try to share some of the information in next few slides with all of you, introducing what BIREC does and how it integrates with the knowledge generation part and then dissemination and bringing it to the life for eventual use. So some of you probably know, but just for the sake of clarity, BIREC is a not-for-profit Government of India undertaking under Ministry of Science and Technology, seeded by a Department of Biotechnology. It supports entrepreneurs, startups, SMEs, as well as large companies to undertake high-risk innovation that can lead to development of products and technologies to address unmet need. As we all understand that deep tech requires longer gestations as well as patient capital combined with a lot of niche expertise, understanding of regulatory compliances, the ability to move together with the right ingredients, right raw material selection, and we just heard about a lot of compliances even after it goes into the market for pharmacovigilance perspective. So BIREC tries to provide a facilitation from idea generation to proof of concept to early stage validation, manufacturing, and taking it to the market through a bouquet of customized state-specific schemes that provide access to funding, mentoring support, access to mentors, experts, regulators, IP strategy and management, the capacities where these ideas can be nurtured beyond the research institutions in the incubation centers, and an access to national and global opportunities, including industry connect as well as investors connect. So over a period of time, in last 9-10 years, there has been a footprint across India, being a national enabling agency. This opportunity has been extended nationwide, and talent pool and the knowledge that has been generated across the sector has been utilized by many people. If you compare, let's say from 2012 to 2021, we have come a long way, and the technologies and the ideas that initiated many years ago, they have been able to get disseminated and translated through an interjection which is led by the startup ecosystem. All of you might have heard that India is the third largest country housing of the startups. You might have heard about unicorns getting seeded. We have now 80+ unicorns in the country, and 42 of them have just emerged only in the last year. So that's a significant movement in the knowledge generation to knowledge deployment in the direction. And the allied activities around it, in terms of number of products, the capacity building, that has also increased, particularly in the biotech sector, where these aspects fall in interdisciplinary, the bioeconomy of the country has also been increasing year on year. We are seeing a growth of more than 12% even in the COVID times, and this is poised to grow to 2x by 2025. BIREC supports the activities across different subsectors of biotechnology, including medtech, biopharma, biotech, industrial biotech, clean environment, as well as agri and allied areas. And accordingly, the capacity building is done in consideration of the requirement, specific requirement for each of these sectors. There are now 60 incubation centers which specialize to support biotech related innovations across the country. There are 10 pre-incubation centers called EUA. They are also spread out. And we also have 4 BIREC regional centers, so that the opportunities as well as ability for people to reach out to this ecosystem is can be leveraged across the country through the combined efforts of the stakeholders and partners that come along. So when you look at the combined effort of all this large platform of partners, we are looking at more than 700 products which have actually moved into the market through these efforts, and that's a large number. These technologies are often led and create IP when they are under development. So you can actually see the IP's wealth that has been generated. It is substantial, and that IP strength is leading to translation into tangible products. So unlike many a times when the IP is generated, but it keeps sitting and not utilized or not exploited. So it's a significant evolvement from the past to the ecosystem where it has now moved in the forward way. There are many opportunities which provides, let's say, funding support to ideation. One of the flagship schemes of BIREC is the Ignition Grant that provides 50 lakh rupees at the ideation stage to entrepreneurs and startups, individual entrepreneurs. That has been supported. More than 700 such proposals have been supported over a period of time, and that has led into translation of products from ideation onwards. So you can imagine typically deep tech technologies would often take 5 to 8 years to emerge into get their first customer on board. But the journey is very exciting. As you can see, there are follow-on funding that gets generated by these entrepreneurs and startups, and the overall ecosystem is attracting many more entrepreneurs in the pipeline. Likewise, there are other schemes I will probably touch upon as I move forward. But just to give you a snapshot in this slide, you could see that year on year, there is an increase, regular increase, in fact, of biotech startup seeding in the ecosystem. And we are looking at these numbers to reach about 10-25. And this startup ecosystem is connecting both the academic research institutions, excuse me, the industry network, and the allied enabling environment. And collectively, I think it is harnessing both the knowledge generation as well as dissemination for tangible deployment. We also see that the talent pool, which is spread across the country, is able to reach out to avail such opportunities. And each dot on the India's map on the left side reflects the people, entrepreneurs who are reaching out and submitting proposals to avail different opportunities. And there is an increasing number in terms of gender parity that we are also seeing year on year. Just like I gave you an example of one of the large major flagship scheme, there are funding provisions that empowers an individual entrepreneur or a startup, all the way from fellowships to $1 million for funding support, so that your venture can nurture and evolve into the market. And this is being done at, as I mentioned, stage-specific different schemes that have been customized and evolved over a period of time, very thoughtfully, that what is required at what stage, and integrating industry, academia into this bandwidth. Such schemes which are enabling the technologies to go forward, and also supporting even the large companies, for example, up to phase 3 clinical trial or manufacturing of a pilot batch, etc. So the translation of knowledge which gets moved up into the at a certain stage, that gets facilitated through these endeavors. And being one of the older and flagship schemes, you could see a lot of academia institutes have got benefited. Different numbers of collaborative projects have been supported under these schemes. Likewise, BIP also has led to technology advancements up to pilot stage and validation stage, moving into technology product deployment through industry participation. Here, the industry co-participates in the technology development to take it forward. One particular scheme which promotes faculty entrepreneurship is PACE. I think that would be of interest to the faculty attending or participating in this interaction. Here also, just like VIG, you get sufficient funding support so that your technology can move that milestone to achieve either a validation that can attract industry to take it forward, or if you intend to do a technology transfer in the instead of taking your technology all the way to seed and venture company of your own. So this fits in and covers that area also. Likewise, we have other schemes to promote social innovation entrepreneurship. There are follow-through funding schemes which provides exposure to equity, and it helps attracting the private funding opportunity from angels and venture capitalists, so that your venture, your startup can actually move forward. And there would be, unlike a large company which has deeper pockets and large resources, individual startup or entrepreneurs do not have that. and you would be continuously raising funds to progress from one milestone to the next milestone. So these are essential as you dwell and move into this direction. There are very promising examples. This is just a snapshot that small amount which gets infused has been able to, let's say, about 100 startups got supported with maybe about 35 or 40 crores. They've been able to now generate more than let's say, put together across these two schemes, LEAP and SEED, you're looking at a generation of about 270 crores put together, and their combined valuations are more than 1,500 crores at present, and this is happening over a period of a few years. And as an outset, that it is also culminating into products that are reaching into market. So there are quite a few instances which are very encouraging and evidently promising that can help and encourage more entrepreneurs into this bandwidth. In next few slides, I'll show that it's not about only funding that you need once you initiate into technology development. You also require a lot of other engagements, and that engagement would include industry experts, the ventures, fundraising from private bodies like angels, VCs, different enabling bodies, ministries, departments, where you might want to look at product deployment in case of G2G business or G2B businesses. BIREC being a Government of India organization, it connects with the large number of stakeholders across different public-private sectors, and that entire strength is available to the people who get integrated into the ecosystem. So that's a powerful network. Besides funding, there is a great emphasis to provide technical expertise through trainings, workshops, regulatory understanding, understanding of how to manage and develop a strategy for IP wealth management. So that also gets integrated and supported through a large network of nationwide footprint of BIREC. We also provide access to international ecosystem so that the product that is getting developed must have an understanding of global competition so that it can beat the competition, as well as it can engage in markets other than India too, because the unmet needs are probably similar across the geographies. And you would realize that India being such a large platform where you have unmet needs, requirement for, let's say, high resource rich provisions to extremely low resource settings. So, and the unmet needs are being catered across this dimension and the two extremes by the entrepreneurs and startups that are venturing into this activity. So we definitely connect, and there is a direct relevance for access to the markets and understanding of global regulatory and compliances requirement. Global Bio India is another platform where the entire biotech ecosystem gets represented from India to the outside world. And the two editions in 2019 and 2021, in each edition, there were 40 plus countries participated, and very encouraged to see where's India's ecosystem is evolving, and can India become global innovation hub for that matter? I'll give you a couple of examples here of ventures, startups supported that have moved into market now, and across the subsectors of biotech, and they are now in market and commercializing their ventures, and these companies are moving forward. Some of these examples also include first-in-class inventions, discoveries, and that has led to new novel products that are either under development or have reached to market or engaged with different industries that have made India's ecosystem, validated the strength of India's ecosystem, not only at national level, but at the international platform. If I would encourage you to visit Virex's e-portal, biotech-solutions.com. It showcases about 220 such technologies which have reached the market, and maybe those could be case studies that you want to follow just to get an idea how are different endeavors reaching to the market. So each of these successful startups in the market, they have been elaborated in this website. This provides basically exposure to at the national level for individual startup as well as at the international platform. This slide suggests that the larger infrastructure capacity building activity is also undertaken, which is thematic and scheme specific, just like for nanotechnology, like biopharma, pharma, and medtech devices. There are specific high-end infrastructure that is getting developed and being utilized at the national level by the startup ecosystem and research institution, entrepreneurs, as well as SMEs or companies who are intending to do high-risk innovations. And you would notice that particularly in the COVID times, the new names have emerged, and they would become household names going forward. I would assume some of you would have used the home diagnostic kit of MyLab. There is Huwell Life Sciences, who has now established a full-fledged manufacturing facility for diagnostics. AMTZ MedTech Zone has been producing diagnostic kits, ventilators, as well as scanners. You would have read today's newspaper that mRNA-based technology platform vaccine is now entering in phase 3 and looking for regulatory approval. So a fourth candidate from India's portfolio of vaccines against COVID successfully moving forward. This only has happened, and there are some more examples that from the young startups and entrepreneurs across the requirements that could be met, and we could address the challenges of COVID through these endeavors. I think this reflects collectively the strength of the ecosystem, that India's innovation ecosystem has been able to take up this challenge and actually respond to it. Unlike many countries who have not been able to handle COVID pandemic consequences, India's ecosystem, and given the non-developed status of our country, we have still been able to handle this large challenge and a sudden challenge that is imposed on the ecosystem in the country. And the biotech ecosystem has been able to stand tall and respond to this. So these are very encouraging instances, but few, I would say. Going forward, I think with the growth of the ecosystem, we can all anticipate that with participation of budding entrepreneurs, let's say from your university, participating in this endeavor, the faculty, your engagement with industry, enablers, just like this particular seminar where you have brought in speciality expertise from across the ecosystem. I think everybody's effort collectively adds strength to this ecosystem, and the opportunities are enhancing and getting added through collective efforts. So I would encourage all of you to seriously undertake the technical expertise that you're gaining while studying in the university, in the college, and taking maybe some of you might want to engage into further studies, get into institutes, research institutes where you want to further dwell into some activities. So some may move from not from research activities, but from marketing, sales, managing large portfolios, managing other aspects of the ecosystem. So still, if you think that there is some convergence that that can keep you connected to the growing ecosystem, that would be very encouraging. So I am very hopeful that we have bright future ahead for many discoveries, many innovations emerging out of India, and probably so would be you. Thank you so much for having me here. I'll stop my screen share here. Thank you.
Thank you so much, Dr. Manish Diwan, for covering the aspect of entrepreneurship in the development of these technologies. And one of the features which you have highlighted, that the spend of the money, like something 130 lakhs, which is giving the product which is worth 270 crores and the valuation of 1500 crores. So this generates that how through this entrepreneurships or through this technology or through even the encouraging the entire ecosystems, the products which is being developed for the mankind and was able to came into the market. And I think very beautifully probably it's been covered that how important is all the stakeholders, that once you have a seed fund and once you have encouraged, and one of the examples we have are in in our university, Delhi Pharmaceutical Research Sciences University, as a Bioness Center, which is supported by the BIREC. And there are about 16 of the incubators which has been working. So definitely, and the various grants which is available to the each individuals, and in fact to all those who are attending, probably looking into the website will be very, very useful, because that seed money will help you and the ecosystem specifically. What Dr. Manish mentioned is that that you have got access. You don't need just your research, but there are a lot of other things which has been required to bring thing into a market, to bring together the scientists, the industry, the angel investors, the other regulatory bodies, to bring any products into this market. So I think for the wonderful talk, and thanks, I think my co-chair, Dr. Majumdar is here. So I think probably you can see that, anyone who is attending offline, if there is any questions, probably, you can encourage them to take that. I don't see anything in the chat box.
Thank you for your talk. I feel privileged that that this should be, oh my God, 20 years back. Now, telling me so many new things. That's very good. And I ask all my students whom I am teaching now, tomorrow, after creating a seat be there. And I will be sitting here, teaching, right? Okay, Manish, I will ask you a simple question. Do you have any simple product promoting like a sanitizer? Because I could not see most of the things sitting over here. Say a simple thing like sanitizer, which is basically a 70% ethanol, will sanitize anything because it is bactericidal, it is cidal, bactericidal, fungicidal, biocides. Aspires to it?
Sir, probably I could not get the complete question, but I could hear that you were referring to a sanitizer like a product. Let me tell you, during the COVID times, even we had very limited availability of such a low-tech solution, sanitizer. So during the COVID time, we could see upsizing of manufacturing of ethanol by several folds by various industries. There were several startups and entrepreneurs who created sanitization solution. For example, digital AI-led sanitizers, the dispensers, the WHO recommended 20-second protocol, for instance, that you put your hands in, it would guide you that how you need to distribute the sanitizer on your hand and keep it exposed to 20 seconds so that the sanitization activity gets completed. Happy to tell you that such a system was developed and we were able to support that, and that automated sanitizer solution right in the beginning, if you could recall the COVID times, that solution was installed in 11 airports when the airports were getting ready to for the passengers, and there were solutions, similar solutions then get deployed at different ministries, departments. Even by BBT had also used the same solution till you got the large tanks, no, etc. That just dwelling further on it, the same kind of sanitization activity was required in ICUs. And now in ICU, you can't plug in, like you can bring in any bottle inside, etc. So you need a right kind of a refill so that it you bring in a just like a saline bottle. When it finishes, you have a saline ready-made and you just align with the dripping line. Likewise, the sanitizer pre-filled bottles need to be aligned into the dispenser. Then only it kind of is acceptable in the ICU regulatory requirement. So these were finer details, but such solutions were provided and developed by the startups in very short time, and those were also successfully that was the requirement of that time and was suitably addressed by the startup ecosystem. So yes, you are right. Both low tech as well as deep tech is a requirement that that addresses a particular challenge at a particular time. Both are
Thank you. Thank you so much, Dr. Diwan. I think you have very well answered these questions, and several of even the bio incubator in our university was able to provide that solutions, which was being used and also applied in DRDO. So, from what you have rightly mentioned, that from low tech to a high tech, and the things like a MyLab, which has again been sponsored, which we are every day now using it, especially in the third wave is being used, is also a result of that, such innovations being in this field of the health sciences. I think I'll we have now we can take up the next speaker, Dr. Diwan.
Yeah, but over to you, ma'am. Dr. Diwan, Anupama Diwan. Sorry.
Yeah, Anupama Diwan. Yeah.
I
Right,
can see Dr. Bansal here. Hello, Arvind.
Yeah.
How are you?
Yeah,
Manish, how are you?
Yeah, I am feeling really very nostalgic. I think one thing has been common, like Arvind, Manish, myself, Dr. Majumdar and Anupama, we both are from the same institute's College of Pharmacy alumni. So,
Yes.
it's a chance or coincidence that, in this platform, probably, all of being together and, dwelling upon the very important thing.
Yeah,
About
thanks for bringing us together.
Yeah.
Greatly appreciate.
Right. Yeah. And it's always pleasure to see Dr. Arvind Bansal, who has been in NIPER and probably, not here, internationally, also worldwide. I keep hearing his name because he is involved deep in science. All you talk about this nanotechnology or the drug delivery, he has been pioneer and he has been working specifically, in this field. So probably, it will be very good. We all talk about a major challenge, facing the healthcare industry is the human body's inability to, sometimes absorb the entire doses of the drug. So this is where, the nanotechnology comes into the picture. And, and specifically, it can be used to transport the drugs to a specific cells of the body, which not only ensures the precise treatment, but also reduces the chances of failure of the rejections. So he's been working, definitely in this field, and several drug therapies, medical devices at the nano level, ultimately ensures a higher level of accuracy in the medicines. And this technology has been leveraged on broad applications. We talk about diagnostics, also the medical devices, also in the nanomedicine. And with this rapid advancement of this nanotechnology, the scope we say is endless. But again, with the high rate of a success, there are a lot of challenges and a lot of hurdles as well. So the more research is definitely been going on, because probably, in the initial years, we are very enthused and looking into the plus points. But, as more and more research has been happening, and when we are seeing the long-term impact of this nanotechnology and a lot of, the environmental applications, so a lot of policy makers, and then, a lot of other things which also needs to be dwelled into. So I'll ask probably the Dr. Anupama and from their sides to read your bio profile, brief profile before, proceeding it to hear from you. We are very eager to hear from you. Dr. Bansal.
Now we will move on to the fourth speaker, Dr. Arvind Bansal, Professor of Pharmaceutics, NIPER, Mohali, Chandigarh. Dr. Arvind Kumar Bansal is currently Professor and Head Department of Pharmaceutics at National Institute of Pharmaceutical Education and Research, SAS Nagar, Punjab, India. He earned his M.Pharm in 1988 and PhD in 1993 from University of Delhi, India. Professor Bansal had worked as senior scientist and group leader in JK Pharmaceuticals and Pharmacy Research Laboratories for 8 years. Therein, he conceptualized, evolved formulation strategies, developed and transferred the technology to production shop floor for NCEs and generic drug products. Professor Bansal joined NIPER in 2000 and developed expertise in various areas. Now I would like to invite Dr. Anupama Diwan to welcome the speaker with green certificate.
It's so nice to have you over here, sir. I really welcome you with folded hands, Dr. Arvind Bansalji, for accepting our request to be a speaker today. So we are looking forward for the time when you can personally, after this pandemic era, can come to the university. But today, on your behalf, we will be planting a tree in our herbal garden. Please accept our gesture, sir. Thank you, sir.
Good afternoon, everyone. And first of all, my sincere thanks to the organizers of the event. Especially, I was in touch with Professor Majumdar and Dr. Anupama Diwan for the event. So thank you very much for inviting me again to this platform. And I'm really happy to see some friendly old faces from our school, college days, and from the days at Ranbaxy. So I see Manish there, Professor Popley is there, and I think in audience, Professor Majumdar and Dr. Anupama Diwan are also there. So the topic that I am going to talk, in fact, has been sent to the organizers as a recorded presentation. I'll just briefly give you the background of what I would be talking about. So it's a nanotechnology enabled platform technology that we have developed at NIPER SAS Nagar to address the poor aqueous solubility of the compounds. Interesting aspect is that the initial lab scale proof of concept was established with the funding from Department of Biotechnology, and subsequently the scale up at the industrial site was done with the funding received from BIREC. So what Dr. Diwan was talking in his presentation, in fact, has been helping a lot of scientists. And with 2 grants in a scheme called BIP, we had scaled up this technology with our industry partner, Messrs. Windlass Biotech at Dehradun, to a kilogram level. This has been a very challenging and a satisfying experience for us. It is extremely difficult to take up technologies from academic institutions into the industrial arena. There are a lot of points that have to be taken care. There are issues related to reproducibility of the data, and there are obvious challenges during the scale up. So in my talk today, I will be discussing a few fundamental things about poor solubility and how nanocrystals are helpful in overcoming them. And then I'll be moving over to the specific technology platform that we call as NanoCrisp, and how it helped us in achieving the objectives of drug delivery. So I request the organizers if they can kindly play the recorded presentation that I had shared with them. Poor water solubility is a very significant challenge in pharmaceutical development. Various tools and formulation technologies have been developed to overcome this issue. One of the techniques that has been found to be industrially viable is drug nanocrystals. Today, I'll be talking about drug nanocrystals in general, and then specifically about our technology platform that has been developed at NIPER SAS Nagar. This technology platform is called NanoCrisp. The contents of the presentation would include introduction. Then we'll talk about the NanoCrisp platform. We'll take a case study of celecoxib nanocrystalline solid dispersion. Thereafter, we'll spend some time understanding the molecular mechanisms responsible for generation of nanocrystals during the NanoCrisp technology, and then finally summarize the talk. Low aqueous solubility may lead to low and variable oral bioavailability, and this pictorial shows the extent of poor aqueous solubility in the marketed drugs and in the pipeline drugs. The BCS class 2 drugs, which are having poor water solubility, are almost 30% in the case of marketed drug, and they have risen to 70% in the pipeline drugs, which means the new molecules that are coming into the drug development pipeline are becoming increasingly poorly water soluble. Enhancing aqueous solubility, therefore, remains a high priority area as it has strong clinical and commercial significance. The strategies that are available to improve low aqueous solubility are diverse in nature and include use of co-solvents, lipid-based systems, surfactants, cyclodextrins for formation of inclusion complex, salts that are only useful in the case of ionizable drug, particle size reduction, use of metastable polymorphic form, designing of co-crystals, and amorphous solid dispersions. Co-solvents will only be useful in the case of liquid formulations and have no significance in oral solid dosage forms. Our talk will be concentrating on the particle size reduction, which can be done either up to the micron size or to the nanometer size. Our emphasis would be the drug nanocrystals, which are in the nanometer size. Drug nanocrystals are defined as crystals with a size in the nanometer range, that is between a few nanometers to 1000 nanometers. In contrast, microparticles possess a size of 1 to 1000 microns. These drug nanocrystals, when incorporated into the dosage form, predominantly consist of the pure API, which means we are not using any matrix or any excipient in large quantity for generation of the nanocrystals. The only additional excipient that we use when we generate nanosuspensions are polymeric or surfactant stabilizers, which keep the nanocrystals in the nanometer range. We'll now discuss the mechanisms that are responsible for enhanced solubility and dissolution kinetics from the nanocrystals. First, let us look at the enhanced dissolution rate. The first and foremost reason is because of the reduction in the particle size, there is a huge increase in the surface area, because of which the dissolution kinetics enhances. The other reason is the decrease in the diffusional path length. Here we have shown that in the case of micron size crystals, the diffusional path length is longer than that is there in the case of the nanocrystals. The other reason is that the saturation solubility increases in the case of drug nanocrystals, and if we see the Noyes-Whitney equation, there is a decrease in the value of D. There is an increase in the value, there is an increase in the value of A, which is the surface area. D is the diffusion coefficient. H is the diffusional path length or diffusional distance, which decreases, and there is an increase in the saturation solubility. And because of this, there will be an enhancement of the dissolution kinetic, which is given by dC by dt. There is always, there is also an increase in the apparent saturation solubility when we use the drug nanocrystals, which is governed by the Ostwald-Freundlich equation. According to this equation, the dissolution velocity from the smaller nanocrystals is faster, which leads to the shifting of the equilibrium towards the solubility side, and the drug gets dissolved up to a concentration which is higher than the saturation solubility. We have to keep one thing in mind, that the increase in the apparent solubility is only visible when the size of the nanocrystals become small, that is below 100 or 50 nanometers. Another important thing to keep in mind is that the increase in the saturation solubility is only up to 1.2 to 1.5 times, and there is not a very high increase in the apparent solubility. But the dissolution kinetics is significantly enhanced in the case of drug nanocrystals. An additional mechanism which is at play during the improvement of oral bioavailability from the nanocrystals is the enhanced mucoadhesion. These nanocrystals are able to create more contact points with the mucus, and they create a kind of depot which helps in release of the drug for a longer period of time. There is no research on the evidence of direct uptake of drug nanocrystals into the systemic circulation. Transcellular uptake majorly occurs through enterocytes and M cells of the pair patch. Transcytotic capabilities of enterocytes is limited, and M cells are present in very small proportion in the total intestinal surface. The level of uptake and to what extent it helps the oral absorption is still suspected. So the main mechanism responsible for enhanced oral bioavailability from the drug nanocrystals would be increase in the dissolution kinetics and partial increase in the saturation solubility. This is a decision tree which has been proposed by Pfizer company for the selection of the most appropriate technology for enhancement of water solubility. 3 technologies are considered when we have a poorly water soluble drug. First one is the nanocrystals. Second is the liquid filled capsules containing lipidic systems. And the third one is the amorphous solid dispersion. Based on the log P value and the melting point and the dose of the drug, these are the areas in which a particular technology is useful for a given drug molecule. For example, if you have a log P from -2 up to 6 and a melting point greater than 200, then only drug nanocrystals are the viable technology for enhancement of water solubility. But in this zone, both nanocrystals and amorphous solid dispersions can be used. And this is the zone where all the 3 technologies, including nanocrystals, lipidic systems, and amorphous solid dispersions can be used. There is a small zone of low melting point and high log P value where only lipidic systems are useful. This diagram is when the dose is less than 100 milligrams. So if you look at the unique zone where only the nanocrystals are useful, then we can see that it is the case for high melting point drugs having a low log P up to a medium to high log P. There are a lot of products that have been commercialized using the nanocrystals, and this table captures the examples of such products. Emend is a product for aprepitant, similarly for magastrol acetate, sirolimus, fenofibrate, and there are so many other drugs for which the nanocrystal based technologies have been used. Top-down technology using media milling is the most common way for generating the nanocrystals and using in the oral solid dosage forms. Other technologies like high pressure homogenization and bottom-up technologies like precipitation have also been used for commercialization of the nanocrystal-based products. There are 2 approaches to generate the drug nanocrystals. One is called as the top-down, where the bigger crystals are broken down into smaller crystals till you reach to the nanocrystal size. Top-down approach is the most commonly used one, but it has some limitations. Like there could be possible decomposition of the drug because there is a very high energy input during the size reduction. It may lead to residue or impurities from the milling media. It cannot handle salt forms, hydrophilic drugs, and drugs having low enthalpy values. Limits posed by the crystal lattice parameters like cleavage plane can sometimes limit the amount of size that can be achieved, or the particle size can be achieved only up to a limited extent. It involves use of surfactants for stabilization of the nanosuspension and uses long processing times. The other approach is called as the bottom-up approach, which also has some limitations, like it can lead to organic solvent residue. It is not universally applicable. and there can be undesired compound recrystallized to the crystalline state. This is the pictorial depiction where the molecular solutions lead to the crystallization and formation of clusters followed by nanocrystals. That is why it is called as bottom-up approach, because we are starting from the molecular level to the nanocrystal size. All these limitations indicate that there is a need for an alternate technology. So there is some space where new technologies can also play their role. And here comes the role of the nanocrisp technology, which was developed at Hyper SAS Nagar for the generation of nanocrystalline solid dispersions. Nanocrisp is a novel bottom-up spray drying based method to generate solid particles containing API nanocrystals dispersed in non-polymeric excipient. This is the pictorial depiction of a single particle which can be in the range of 0.5 to 20 microns, and you can see the green color boxes are that of the drug nanocrystals, while the yellow ones indicate the small molecule excipient. The drug nanocrystals are in the size of 10 to 1000 nanometer. So each particle consists of a number of API nanocrystals in the range of 10 to 1000 nanometer dispersed in the matrix of the excipient. This is the pictorial depiction for the generation of the drug nanocrystals using nanocrisps. This is the spray drying of the droplets, and this is the single droplet that had been shown. Gray color circles are that of the solvent. The yellow ones is the excipient, and green one is the drug molecule. So this is the initial droplet from where the solvent starts evaporating during the spray drying process, and all the solvent will be then removed from the droplet, leading to the formation of a single particle wherein the drug nanocrystals are embedded in the matrix of the excipient. Now we'll take a case study of the nanocrisp technology, and we will be talking of celecoxib, mannitol, sodium lauryl sulfate system, where the proportion of the ingredient is 30:65:5 respectively. The generated nanocrystalline solid dispersion was found to be crystalline in nature, and this is the melting point of the drug, followed by the melting point of the mannitol. The spectrogram indicated the crystallinity of the nanocrystalline solid dispersion sample, because there were good quality diffraction peaks observed for the product. These were the spray drying parameters that were used for the generation of the nanocrystalline solid dispersion. This is the scanning electron microscopy photograph of the primary particles, and this is the transmission electron microscopy of a single particle. This is a single particle in which this dark colored spots indicate the nanocrystals of the drug that are present in the single particle of the nanocrystalline solid dispersion. Using the dynamic laser scanning microscopy, the D90 value was found to be 470.33 nanometers with a PDI of 1.0. The PDI was on the higher side because of the presence of some bigger particles in the sample. So SEM, TEM and DLS indicate particle size of the NCSP sample. Dissolution studies were carried out at pH 11.7, which is able to discriminate between the products, and there is a comparison of the nanocrystalline solid dispersion with Celebrex, which is the marketed product of celecoxib in manufactured by Pfizer. There is a very significant increase in the dissolution kinetics of the nanocrystalline solid dispersion over the marketed product, that is Celebrex. After this, we had also carried out the oral bioavailability study in the animal model, and we found around 4 times improvement in the AUC values. The data has not been shown here. And there was a decrease in the Tmax, which means the Tmax was achieved faster, and there was an increase in the area under the curve. Now let us look at the molecular mechanisms that are responsible for generation of the nanocrystals during the nanocrisp process. The solid form that is obtained during spray drying could either be amorphous or crystalline. We are interested in generation of the crystalline end product, and we don't want the drug to be in the amorphous form. The reason for this is that the amorphous form by spray drying is a patented technology of some other company. So we wanted that our drug should be present in the crystalline form. So we looked at the thermodynamics, kinetic, and molecular mechanisms that are involved in the generation of nanocrystals during the nanocrisp process. First of all, we looked at the solid form behavior of the API. The Oswald step rule says that crystal phase that nucleates first is not the phase that is thermodynamically stable at that temperature and pressure, but rather another metastable phase that is closest in free energy to the parent phase. In fact, in the case of celecoxib, we see formation of the amorphous form, and in the case of curcumin, we saw the formation of the metastable polymorph, which is in accordance with. Out of this understanding is that API is first precipitated in the amorphous form from the solution state, which then crystallizes to the nanocrystals. So here we can see, when we see the DSC heating curve of celecoxib, mannitol, NSD without the annealing step, there is evidence of presence of amorphous celecoxib. So this is the place where we zoom in to find out the glass transition temperature of the celecoxib amorphous form as 57 degrees Celsius. However, post annealing, we see that this glass transition temperature has disappeared and there is no event of glass transition, and all the drug has now converted into the crystalline form. Annealing is the phenomena of heating the drug at a temperature which is slightly higher than its glass transition temperature, and by increasing the molecular mobility, the amorphous form is then converted into the crystalline form. So the conclusion from this study was that celecoxib initially comes out in the amorphous as well as the crystalline form, and to get completely crystalline form, we have to anneal the sample for some time. The crystallization from the amorphous form is dictated by the classical nucleation theory. Crystallization occurs easily above Tg in 2 stages, that is nucleation and crystal growth. Nucleation happens at a temperature close to Tg, and crystal growth happens at temperature towards the melting point. So this is the Tg. So around this, there will be tendency of nucleation, while this is the melting point or Tm. So towards this end, there will be tendency of crystal growth. This is because of the fact that Tg and molecular mobility is related. If higher the temperature, the molecular mobility will be higher. So the inlet temperature during the spray drying should be between the nucleation range and the crystal growth range. So this is the overlapping zone where the nanocrystals are expected to form, because this is midway between the nucleation and the crystal growth. If the temperature is on the higher side, the crystals will grow into bigger crystals, maybe into the micron size. While if we are at this stage, there will be formation of small nuclei which will not grow into micron size crystals. So the desirable aspect during the spray drying is formation of a large number of nuclei that do not grow into micron size crystals. Inherent crystallization tendency of the API also plays an important role in the generation of the nanocrystals. Aspirin, which was investigated as another molecule in our lab, has a very high crystallization tendency, whereas celecoxib has comparatively slower crystallization tendency because of high entropic barrier to crystallization. The higher entropic barrier of celecoxib may be attributed to its ability to exist in larger number of molecular conformations because of the 4 rotatable bonds that are present in the chemical structure of celecoxib. So the practical implications of this fact during the nanocrisp technology are that aspirin forms nanocrystals in the presence of mannitol in the temperature range of 30 to 100, while celecoxib forms nanocrystals only above 120 degrees Celsius, and the Tg of celecoxib in presence of mannitol is 57 degrees Celsius. Crystallization tendency can also be enhanced by including a crystallization inducing excipient in the formulation. Crystallization inducing excipients enable crystallization of the drug from its amorphous state during the spray drying in the nanocrisp technology. Now we look at the functionality of the excipients and their crystallization inducing potential, which is important for generation of nanocrystals in the nanocrisp technology. So how does the crystallization inducing excipient help in the generation of nanocrystals? The first mechanism that we found out was plasticization or reduction of the Tg of the amorphous API. So at a given temperature, system with lower Tg has a greater tendency of crystallization. And this was exemplified by the example of hesperidine and mannitol system. Amorphous hesperidine has a Tg value of 72.9, while in the presence of mannitol, the glass transition temperature reduces to 59.2. So decrease in the Tg increases the molecular mobility of amorphous hesperidine at a given temperature. Thus, mannitol here acts as a plasticizer for hesperidine, enhancing its molecular mobility, leading to crystallization of amorphous hesperidine into nanocrystals. For the plasticization to take place, there is a requirement of miscibility between the drug and the excipient. Miscibility between the drug and excipient is necessary to plasticize the drug and accelerate its molecular motion. And miscibility can be evaluated by looking at the solubility parameters of drug and excipients. Closer the values, that is less than 7 of the solubility parameters of drug and the excipient, indicates miscibility between the drug and the excipient. As you can say, in the case of hesperidine and mannitol, the solubility parameter is quite close to each other, and these two systems are miscible, which leads to plasticization and reduction of the glass transition temperature. However, in the example that we have been discussing of celecoxib and mannitol, there is a large difference between the solubility parameter of celecoxib and mannitol, and these two ingredients are immiscible with each other, which means plasticization or reduction of Tg will not be a mechanism in the case of celecoxib-mannitol system. So there is an absence of plasticization in the immiscible systems, which is exemplified by the case of celecoxib and mannitol system. The amorphous celecoxib has a Tg of 57.5, while in the celecoxib mannitol physical mixture, the Tg remains around 57 only, which means there is no substantial decrease in the Tg value. So this led us to ask a question that are there any additional mechanisms at play since there was no plasticization taking place in the case of celecoxib, and a single TGE was observed despite immiscibility because only celecoxib is in the amorphous state and mannitol is in the crystalline state. So we then went ahead and looked at the additional mechanism that may be responsible for generation of the nanocrystals. And the answer came in terms of the primary heterogeneous nucleation and its role in generation of the nanocrystals of the drug. So we did a study on the hot stage microscopy, wherein we gave heat-cool heat cycle to the mixture of celecoxib and mannitol. So this was the cycle 1, where mannitol and celecoxib were melted together as a physical mixture. Cycle 2 was the cooling cycle, and mannitol, as we know, has a higher crystallization tendency, and there is appearance of crystalline mannitol during the cooling cycle. However, it does not lead to crystallization of celecoxib because celecoxib is relatively difficult to crystallization. During the third cycle, which was the heating cycle, after heating, cooling, and then heating, there started appearance of the celecoxib crystals at the sites where the mannitol crystals were already present. You can see there is formation of the celecoxib crystal here, which keeps on growing in the subsequent slides, where the temperature is slightly increasing, which means that the crystals of mannitol, which had already formed, provided the primary heterogeneous nucleation site for the generation of the celecoxib crystals. Now let us understand what we mean by this primary heterogeneous nucleation. So heterogeneous nucleation is a surface assisted phenomena, and the rate and extent of nucleation is controlled by free energy barrier for nucleation in the system containing API alone and API along with the crystallization inducing excipient. When the API is alone, the phenomena that take place is homogeneous nucleation, and when along with it excipient is present, heterogeneous nucleation is responsible for the generation of the crystals. The free energy required for homogeneous nucleation is higher than that required for the heterogeneous nucleation. The interfacial free energy between the nucleus of the API and substrate is indicated by the contact angle, that is smaller contact angle between particles lowers the interfacial energy and reduces the nucleation barrier. Hence, the presence of heteronuclei of mannitol provides a surface for nucleation of celecoxib to occur by reducing the free energy barrier. And this was responsible for the generation of celecoxib nanocrystals during the spray drying process. Another mechanism that is responsible for the generation of the nanocrystals is physical barrier to the crystal growth. During spray drying process, when the droplets it at a particular temperature, it absorbs necessary heat of vaporization and the solvent is evaporated. Excipient having faster crystallization kinetics precipitates in the crystalline form earlier than the drug. Precipitated excipient creates a physical barrier to crystal growth of drug, thereby restraining its size in nanometer range. Overall, if we look at the mechanisms, the plasticization or reduction of TG of the amorphous API is one mechanism. Primary heterogeneous nucleation is the other mechanism, and physical barrier to crystal growth is the third mechanism. So the summary of the mechanistic understanding of the formation of nanocrystals during nanocrisp processes, that we start with drug excipient and solvent solution. The solvent is removed. The drug precipitates in the amorphous form. The plasticization of the drug by excipient leads to annealing, and for this, it can lead to formation of the nanocrystals. However, the requirement for this is that the drug and the excipient should be miscible with each other. If there is not formation of the nanocrystals early on, during the spray drying itself, annealing will take place, and other mechanism, that is the heterogeneous nucleation by the excipient and physical barrier to the crystal growth of the drug by excipient, will contribute to the formation of the nanocrystalline solid dispersion of the drug. So this is a mechanism which is specifically at play during the nanocrisp technology. And in a series of studies, we have identified these molecular mechanisms, and all of these have been published in the peer-reviewed journals as well. Now, concluding for the entire talk, poor aqueous solubility remains an important biopharmaceutical challenge. Nanocrystals improve solubility by enhancing surface area, decreasing diffusion path length, and increasing mucoadhesion. Top-down and bottom-up approaches can be used for the generation of the nanocrystals, as is evident by the commercialized products. Nanocrysp is a novel bottom-up technology for generation of nanocrystalline solid dispersions. Plasticization of the amorphous API, heterogeneous nucleation, and barrier to crystal growth are the molecular mechanisms responsible for generation of nanocrystals during nanocrisp. In conclusion, we can say that the nanocrystals is a robust platform to improve the delivery of poorly water-soluble drugs, as they can help in enhancing the dissolution kinetics and apparent solubility, leading to the improvement of oral bioavailability. So with this, I come to the end of this presentation. If there are any questions, I'll be happy to answer those. Thank you very much for your attention.
Thank you, Dr. Arvind Bansal. It was, very clear presentations, specifically, on one of the technology, and it's a clear example that, it's not, something which is theoretical, but, there is something which has been developed in the research institutes and jointly, it's been scaled up and is being used for the industry. So I'm sure, there will be a lot of questions. So first we'll take up the questions and then we'll conclude this session. Is there any questions? I can see the raised hands, or Dr. Majumdar can help, to bring that structure. And in fact, one of the observations which I wanted to say is that, although, this technology we had been used in the drug delivery, but there are, a lot of research which has been going on internationally as well, on different types of the approaches which nanotechnology is being used. And that is, one of the stealth or a camouflaging kind of a technology. And especially, when we talk at the international level as well, there are, not only the drug delivery, but the biomaterials science. There is a lot of advancement which has been happening. And he has been mentioning that the primary goal of these research is to specific drug targeting and delivery, and reduction in toxicity, side effects, the safety and biocompatibility. And also you mentioned about these things, but similarly this technology is being also used, I think, in nanoparticles as antimicrobial agents for the treatment of wounds. There is also a quantum dots which is being to identify the location of the cancers in the body, and nano devices that also directly reaches the cancer cells to minimize the the damage to the healthy cells. There are nanothermal devices to destroy the cancer cells and minimal the damage to surrounding healthy cells, nanotubes and bone replacement and nanobiosensors. So the beginning is that the drug delivery and the pace of this new drug delivery is especially in the biotechnology. And as Dr. Bansal mentioned, there is more and more issues. And he very relevantly brought that, in the marketed product, it's 30%, but in the 70% of the products which is in the pipeline, there is an issue of the solubility or the BCA. So they fall under that category where you need to apply some technologies to make it to to reach at the molecular level, and to have a predictive and the preventive products which is being available. And this is this is one of the examples which he applied it to the drug. But similarly, the same technology or the same platform technology is applied to a lot of biomaterials as well, and to make such kind of a nano biomaterial. So you call it in the name of the nano conjugates or some of these agents or is the product, but definitely these developments are being very useful. Dr. Majumdar, can you help us to take some few questions, if there is anything in the audience? I think we are closer to the lunchtime, and I don't see any kind of a question. So thank you so much, Dr. Bansal. And it's really encouraging, and I think directly, especially in the research institutes, the students who has been attending will be very motivated to see that at the micro level, that how they can work on these technologies and bring that in that. And there is also a power of not just the funding, but also bringing it together. And very nicely you mentioned that you started this with the DBT funding, and then scaled up with the BIREC funding. And in the previous speaker, Dr. Manish Diwan, he captured that what kind of a funding is been available. So in total, this segment tells about that, what kind of stakeholders has been required, what kind of funding is required, and what you can achieve, through that. So it is not very far-fetched, and thank you so much, to bringing it to everyone being there. I personally enjoyed, the learning about so much about this technology. Thank you.
Thank you very much for the opportunity, and I am sure that this can motivate some of the students. That would be a wonderful outcome of the talk. So thank you very much.
Now I want to invite Majumdar sir on the podium to ask.
Thank you for your nice presentation. I have one or two questions. In the isolation study, why did you take up the pH around 11.7?
Sorry, I could not hear it clearly. Can somebody repeat it for me?
What I want to ask that while studying the dissolution, why did you keep the pH at around 11 or 11.7? Because in our GI tract, the maximum pH available would be equal, that will be somewhere around 8.
So that's a good question. The drug molecule celecoxib is a weakly acidic drug with a pK value of 11.1.
Okay.
So the dissolution medium that is suggested in Office of Generic Drugs website, that is OGD medium, is pH 12 medium. But in our studies, we found that pH 12 medium is non-discriminatory for the particle size of the delivery system. So we did a series of studies to find out a pH value where we are able to discriminate the dissolution from different size of the particles. And we ended up with this value of 11.7. So this has no in vivo relevance, but this was used as a medium for screening the prototypes from a performance perspective.
Okay, done. Fine, that's fine.
Now I would like to invite Dean Dr. Anupama Diwan ma'am to felicitate our guest with certificate of excellence.
I think this is a very noble initiative of planting a tree in the university campus. I think it will be a lifelong memory.
Yes, sir. Thank you very much. And this is just a token of appreciation to you, sir. And would love now again my request when things becomes normal or when you plan to visit Delhi, we are with folding hands, always be at ASU to welcome you. Thank you very much, sir, for coming.
Thank you very much. Thank you.
Thank you.
Thank you so much, ma'am, sir. Now I would like to invite Dr. Havinder Popri ma'am to receive the certificate.
I'll do it myself. First of all, I would like to extend my warm thanks to Dr. Manish Diwan for being the speaker today and kindly receive this gesture from all of us. Thank you very much, sir.
Thank you, Dr. Parna. It's perfectly fine. Yeah.
You are my mentor and guide also. Thank you very much for accepting our invitation.
Thank you. Thank you. Thank you.
Now I want, thank you so much, ma'am. Now I want to call Majumdar sir and Mukesh sir as well as team ma'am on the stage to receive memento and certificate from the team ma'am. Thank you. Now next I want to invite Gaurav sir to receive green certificate from our team ma'am.
Dr. Gaurav Jain and Dr. Aparna ma'am would have been kind enough to be the jury of abstract being presented by our students. The online evaluation and offline evaluation will be conducted during the lunch hour. I'm really thrilled to have Dr. Gaurav Jain of online available with us and Dr. Aparna offline available with us. May I request Dr. Majumdar to come on the stage and felicitate Dr. Aparna and our gesture to Gaurav, Dr. Gaurav online. Please accept it. Dr. Aparna, please come on the dais. Dr. Majumdar sir, please come on the dais. Yeah. Dr. Aparna Raghavan, she is the principal, Advanced Institute of Pharmacy, Palwal. She, her faculty, and the students have participated. Thank you, Dr. Majumdar and Dr. Aparna. In continuation, I'm not leaving the dais to my his fellow comparer. I'm continuing because the lunch is waiting for all of them. So we have today honor to have other faculty members with us. May I request Dr. Majumdar to come on the dais? And we have with us our industry sales, Dr. Yassine. Please come on the stage to receive a token of small gesture from us. May I request Ruchika ma'am to please come on the dais. She is faculty from Advanced Institution, to come and just receive a token of gesture from APJ Satya University. May I request Shivani, a faculty, to please come on the dais to receive a gesture from APJ Satya University. The faculty and students from different institutions are present, and that really makes a difference. Thank you very much. With this, all the dear students who are present offline, I could see a huge gathering here. You can go for your poster evaluation simultaneously, finish it off, and immediately come for the lunch. And we have online that evaluation also going. And after we also have Mr. Chandran and Ms. Hina from Lucknow. Please come on the dais. They have traveled from Lucknow to APJ Satya University. I'm really thankful to you. Both of them, along with their students, have come to really. We welcome you here. Thank you. Mr. Chandran. Madam Hina. Thank you very much. Now we have 45 minutes of lunch break. And online, yes, what I can say is that if you would have been at campus, would have loved to serve a nice lunch, but we will be missing you. But all of us will resume back after 45 minutes very much over here. Thank you very much, and let's the conference and the zeal continue. This is what I said in the morning. The memento should carry on. It should not finish here for the day. It has to continue for years and days together. Thank you very much. We will be back soon.
ओ भूपेश सर कहाँ हो गए? भूपेश सर वहाँ ही है। नहीं यार।
क्योंकि हम लोग को खाना ही नहीं जाना था अंदर यहाँ रखा था मैं यहाँ बैठ गई पहले तो आपके रूम में चले गए थे ना सर हम लोग जो प्रिंट निकालने के लिए रख दिया था हमने और ये सुविनी यहाँ रखे सर
इसीलिए आपको ब्रेक में कैसे करते हैं
हाँ मैं इसमें येलो वाली तरफ करती हूँ बोलो जल्दी आके बोलो
Good afternoon, sir. Good afternoon, everyone. Neeraj, sir.
Yeah, good afternoon. Very good afternoon.
Yes, sir. After this is after lunch session, and the session was quite late, and that's why all the participants are again joining after lunch.
Okay.
So we
Okay.
will
No
be
problem.
starting the session in 10 minutes.
No
So,
problem.
if you can share your PPT and we can see if is it visible or everything we can.
Okay. 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 I will do that. Right. Do you want me to mail it to you? You want me to mail that?
Just share it
I
on
will.
the share screen.
I will. I will do that.
At this
Sure.
time.
Sure. Sure. Just a minute. I will do that. Yeah. So I will do it through share screen. Am I right?
Yes, sir.
Yeah. Just. Yeah. Yeah. Is it okay?
Yes, sir. It is visible.
Yeah. I think right. Is it right, ma'am?
Yes, sir. Yes, sir. Perfectly.
Very good. Very good.
So we are just waiting for all the participants to join. So
Perfect.
yes, sir.
Perfect. So should I keep it on a share mode, ma'am?
Please unshare now, and because we'll share some certificate and everything.
Yeah, yeah. So I will share it again whenever you advise me to do so. Okay.
Thank you, sir.
Sure. Perfectly fine.
Hello. Can you hear me, please?
Good afternoon, Professor Bansi sir. So, my voice is audible. Good afternoon, Professor Bansi sir.
Good afternoon.
Am I audible?
Hello.
Yes,
Yes,
sir.
you are audible. Yes. Can you see me? Can you see me?
Yes, sir. I can see your slide, sir. Sir, sir, इसको आप full view में भी कर सकते हैं. So by slideshow on the pressing by F5.
Yeah, can you see my slide?
Yes, sir. That is fine.
Okay, thank you.
Yes, thank you, sir. So please ensure for now.
Yeah. I wait for your signal.
Thank you, sir. Okay, sir.
Okay. पहले मेरा जॉब है उसके बाद होगा Hello.
Yes, sir.
Can you hear me?
After Neeraj Kumar Chopra, sir. Sir, your time is 2:40.
Starting, yeah. Can you see my slide? Can you see my slide, please? Hello. Can you see my slide, please?
हेलो यस सर यस सर आपकी स्लाइड हमें विजिबल थी और हमने देख ली थी सर आपका जो सेशन है दैट सेशन इज आफ्टर धीरज सर और आपका जो टाइमिंग है सर दैट इज अराउंड 2:40 yes sir so this session is here quite late so after Neeraj sir we will have your session around 3:20 sir. So we are just starting the session.
Okay. Hello.
Yes, sir.
Yeah, you can hear me. I just wanted to make sure that I'm audible.
Yes, sir, you're audible.
Okay.
Good afternoon, all dignitaries and the participants. Now, I, Sana Sita,
and I, Niharika Agrawal, will take over the podium. है समय नदी की धारा, सब बह जाया करते हैं, कुछ संस्थान ऐसे होते हैं जो इतिहास रचाया करते हैं, उन्हीं में से एक है हमारा ये. With this beautiful quote, I would like to invite first session chair Dr. Jitendra Madan. He is Associate Professor in Pharmaceutics, National Institute of Pharmaceutical Education of Research, He is PhD in Pharmaceutical Sciences from B.R. Ambedkar Center for Medical Research and University. And major areas of research include self-assembled supramolecular systems with lysosomes, lysosomes, cyclodextrin, nano and microparticles, and solid lipid nanoparticles. The focus is centered onto the exploitation of US FDA-approved biomaterials in combination with other potential lipids and polymers of the innovative dosage form and drug delivery system in order to improve the bioavailability, stability, safety tolerance, and patient compliance. While a wide range of applications are being taken up, the special emphasis is given to augment the aquacellular solubility and bioavailability of anti-cancer drugs. Moreover, we emphasize on design, synthesis, and development of ligand conjugated stealth nanoconstructs for delivery and targeting of anti-cancer drugs in cancer cells, both in vitro and vivo. Now, I would like to invite Anupama ma'am to welcome our guest with green certificate.
I'm really honored today to have with us a wonderful speaker, Dr. Chopra. He's been, when last session when he has spoken on the topic has been really of immense, knowledge sharing, and it was a great help for the students to understand the basic in such a nice way. We also have with us Dr. Jitendra Madan from Nagpur, Hyderabad, who will be chairing the session, and as I said, just a token of regard, as you guys are online, not present at the campus, so we will be planting a tree in our herbal garden in your name. So this certificate of honor to you, Dr. Jitendra Madan, and to Dr. Dheeraj as well. So please accept it from our side. Thank you very much.
Thank you, ma'am. Thank you, sir. Now, I would like to have Dr. Dheeraj Kumar Chopra, currently working as a Vice President at R&D Department of Aminal Pharmaceuticals, Ahmedabad. He completed his PhD from Shiksha O Anushasana University, M.Pharm in Pharmaceutics from Punjab University, and MBA from Indira Gandhi National Open University. He has over 25 years industry experience, having key skills in product development, analytical development, packaging development, NDDS, parental product, aspect processing. Dr. Chopra is Six Sigma Black Belt certified from American Society for Quality. He has over 30 publications in journals of high reputation and presented his work in various conferences, synopsis, seminars, and workshops. Now, I would like to call Anupama ma'am to greet our guest with green certificate.
Now, I would like to call Jitendra sir on the platform to share a few words. Dr. Jitendra sir.
Hello. Am I audible?
Yes, sir.
Okay. Thank you, everyone, and very good afternoon to all of you, especially Vice Chancellor Sir of FPJ Satya University and Professor Anupama Diwan, Dean School of Pharmaceutical Sciences. And thank you inviting me for chairing the session. As told by the anchor that Dr. Chopra is currently working as Vice President and R&D Department of Aminal Pharmaceutical, Ahmedabad. He completed his PhD from Shiksha Anusandhan University, M.Pharm and Pharmaceutical Sciences, Punjab University, and MBA from Indira Gandhi National Open University. He has over 25 years of experience. Industry having key skills in product development, analytical development, packaging development, generic products, development for regulated markets. Now I request Dr. Dheeraj Chopra for the invited talk.
Thank you. Thanks a lot, Dr. Jitendra, for the kind words. And thanks to the organizers for giving me this opportunity to talk about, to deliberate today. Thanks a lot. And with this, I will like to initiate. Just let me put the slides here. Yeah, are my slides visible now?
Yeah.
Yes, sir.
Yeah. So good afternoon to you all. So for next maybe 30 to 40 minutes, I will talk upon the biodegradable polymer-based particulate drug delivery systems, and I'll be specifically talking about the key factors for polymer selection. How do we select the polymer while formulating these drug delivery system? So here I will just like to emphasize, yes, we talk about particulate drug delivery system, and generally we know these delivery systems by the name of their APIs. We talk about leuprolide depot injection, we talk about triptorelin microspheres, we talk about gossaline implant or so, but we equally important besides the API, yes, which definitely is responsible for the pharmacodynamic activity of the delivery system. Equally important is the polymer in the background, which controls the release kinetics. So that is what, I will be talking about, that background, the polymer background, how important it is. It is like in our day-to-day life also, you see like here, Dheeraj is talking a session here, but there is a lot more in the background. There is a lot background in the times of the contribution of the parents who provided the education, contribution of the spouse, my wife, who ensures that, besides doing her profession, she takes care of the kids, she takes care of the home, so that I can come to the lab, I can learn, and I can give talk here. And equally important, our teachers, who taught me, and that's how Dheeraj is talking today. So with that note, I will just dedicate my talk to my teachers, Dr. Majumdar, Dr. Khar, Dr. Harvinder Popli. I hope, some of those my teachers will be here. So thanks, I take this opportunity to express my gratitude towards my teacher who enabled me, and so that I'm here delivering this talk in front of you. So thank you to my teachers. So with this, I will just move ahead. As we said that, yes, we will be talking about that polymer-based, biodegradable polymer-based delivery system. And before we talk about, what are the key factors for selection of the polymer, what are the critical quality attributes, or critical material attributes of this polymer before we, select them for the delivery system. Let us just have a few words about particulate delivery system. As we know, particulate delivery system means this is small particle, which may be microparticle, which may be nanoparticle, of the polymer where drug is entrapped. And then this polymer, which erodes over a period of time and releases the drug for the drug delivery system to be effective. And this is again a schematic representation of, of a particulate delivery system. You see that drug initially, when a particle it is injected into a tissue, maybe intramuscular, subcutaneous, there is initial release from the surface, and you see here the drug, the red, what color it denotes is the polymer, and encapsulated blue color is the drug. And over a period of time, the polymer comes out and erodes and biodegrades, and drug comes out over a period of time. It may be a period of 1 month depot, it may be period of 3 month depot, and nowadays even like Lupron 6-month depot is also available in the market. So as we move ahead, further just talking about, since most of these polymer delivery systems which are marketed, 90% of them are, ester-based polymers like polylactic acid, glycolic acid, what we call simply PLGA, or polylactic acid, what we call PLA. And what happens, these are being ester-based polymers when their particles are injected into the biological fluids, which are more or less, aqueous in nature. And because of the aqueous environment, the ester bond cleavage happens, and that because of this scission, this cleavage of the polymer chain, the polymer degrades, and gradually over a period of time drug comes out. But how important here is that, how we manage this rate of degradation of polymer, because this rate of degradation of polymer, this chain breakage of the polymer, its mechanism is actually dictating that how drug comes out of that particle, how drug comes out of the delivery system and goes into the systemic systemic circulation, giving pharmacodynamic, desired pharmacokinetic activity, pharmacodynamic activity, and whether depot injection will be a 15-day depot injection or a 1-month depot injection or a 6-month depot injection, that will be controlled by, this polymer. So here it is talking about, simple ester-based polylactic glycolic acid. You see, the long chain becomes shorter chain, it becomes oligomer, and then finally it becomes monomer. You see here the lactic acid, and you see here the glycolic acid, which gets eliminated from the body subsequently. And this is just, a brief, again, schematic description of the hydrolytic bulk erosion of the polymer, because most of these delivery systems which are in the market, as I said, they are 90% of polyester based, that is polylactic acid, the glycolic acid, and their mechanism of action, generally it is bulk erosion of the polymer. There are very few delivery systems which are surface erosion based, but most of them are bulk erosion. So what happens simply, you see, this is the drug particulate, drug particle when where drug is embedded in the polymer, when it is injected into, say, into intramuscular, then there are water molecules in the biological fluid which get diffused into the delivery system, into the particle. And when these water molecules, they get diffused into the polymer, then what happens, because of the degradation of the polymer, that the polymer hydrolysis happens, and you see here the small acid oligomers are formed. When I say oligomers, a small chain or medium chain moieties. And as we further move ahead, more and more acid oligomers are formed. You see these void spaces are created, and then because of this lot of void spaces created, water molecule comes out, taking along with them the drug outside. And this is how, drug actually comes out of the delivery system, and over a period of time, the polymer actually erodes, it becomes, spongy like structure, and the whole drug, entrapped drug, comes out subsequently. And this is, that the scanning electron microscopic pictures of actually the drug delivery system, that is particulate delivery system, over a period of hydrolysis into the aqueous fluid. So this is basically the microspheres put into a in vitro dissolution model, where you see over a period of time how the polymer is eroding actually. And then you see here by the end of 1 month, 4 weeks, lot of void space and that end, which allows the drug to come out, giving it a sustained release action of the drug. And thanks to all these delivery systems, that the knowledge that some marketed products which are there in the market available in the commercial space, this is the first biodegradable polymer based depot injection which was introduced in the market, decapeptyl depot, and it is still there for almost more than 28 years, I believe. And yes, Lupron depot, which is also was the third one. The first one decapeptyl, second was Zoladex, and third is the Lupron depot, 1 month depot and 3 month depot. And this is also polymer based delivery system. And I know many of you who work into this delivery systems, that it takes time to develop these sort of products, and still very few generic such products are there. Actually, PLGA based delivery system, not even a single generic product got approval till date, because of the various complexities. And one of that complexity in this is right selection of the polymer, which we'll talk about. And besides that, there are so many other formulation challenges, scale up challenges, clinical challenges. That is why, people many times, they ask me that why it takes so much time, why you scientists take so much time to develop these sort of delivery systems. Sometimes it takes 5 years, 10 years. There are some laboratories who have been working on, and some, people from outside, they surprise that, why so much time it takes or so. And sometimes, failure happens and all those things, etc. Yes, it happens because of the complexity of the system. And one of that complexity we will talk about, that is right polymer selection. And when people ask me, why is it so challenging, इतना मुश्किल क्यों है, तो अक्सर मैं उनको बोलता हूँ, भैया आप, once you start working on it, you will come to know how challenging it is. Only when you work on it, you realize the challenges into these systems. और मैं अक्सर उनको, मुनीर नियाजी का एक शेर सुना देता हूँ कि उनसे नैन मिलाकर देखो, ये धोखा भी खाकर देखो. वो तो जब काम करते हैं तो ही चैलेंज का पता चलता है. Fair. Well, moving ahead, polymers and depot injection. So when, yes, when we select the polymer for this particular delivery system, polymer for depot injection, so what, the scientists look for? They look for, a non-toxic polymer. They look for a polymer which is biocompatible. They look for polymer, which is having a very controlled degradation, either bulk erosion or surface erosion. As I said, mostly the bulk erosion. And there have been some successful polymers which are based upon which deliveries are available in the market, like PLGA, PLA, even polyanhydride. Even now we have polyorthoester also, but 90% of the deliveries are based upon PLGA and PLA. So with these few words, as further we move ahead, regarding that, biodegradable polymer and just how the chronology was involved, chronology of this biodegradable polymer. So in, actually the need for these polymer were felt long back, and a company like DuPont, they started working on it. DuPont had been working for quite some time. So somewhere in 1960s or so, a lot of research was done on the polylactic glycolic acid copolymer, and first resorbable suture was launched by Johnson & Johnson by the brand name Daxon, which was in 1970. And taking a learning from that, formulation scientists used this PLGA, which was used for making the approved Daxon suture, the biodegradable suture. They worked upon, and in 1973, drug development started with this PLGA-based system. In 1970s, a lot of clinical trials were done for products like leuprolide, triptorelin, goserelin, or so on. And the success actually, first success came by the name of triptorelin, that is decapeptyl by Faring in Denmark. They got the first one, and as I said, second was Zoladex by AstraZeneca in UK, and third was by Takeda in Japan for leuprolide. So this is how, that is just a history behind how polymer-based delivery systems came into the market. Just before we go into the actual CMAs of the polymers, just depiction that what is basically the most important biodegradable polymer used in commercial biodegradable delivery system. That is basically a copolymer of lactide and glycolide, means lactic acid and glycolic acid. So here you see here, the lactic acid moiety, and here you see the glycolic acid moiety. Actually, these are two homologous series actually. So the glycolic acid and the higher homologous lactic acid, where one proton is replaced by the methylene group, and you can say here that this is the lactide moiety. And keep it in mind, because we will talk more about it later. The lactide glycolide, which is a smaller moiety, so naturally more hydrophilic in nature. Lactide, which is a higher homolog, so obviously it is a slightly more lipophilic in nature compared to glycolide. And the advantage of these being, yes, why this polymer got success, because its medical safety was established. It has been used in biodegradable sutures. Safety profile was established. This polymer is biodegradable, non-toxic, and over a period of time, this polymer actually degrades to lactide, glycolide gets, and lactide goes into the Krebs cycle of the body, glycolide excreted in the urine. So it is totally biodegradable. So there is no remnant remains in the body after the completion of the therapy. So this is, some of those advantages. Yes, it has some challenges also on the, and that, but still, very acceptable polymer for delivery systems. So now let us, when we see that the various types of, factors which influence the degradation behavior, of this polymer and dictate the drug release pattern from this polymer. When I say, like, say, polylactide glycolic acid. So there are so many things, but purely from polymer point of view, these are some of the factors. I'm saying not all, but some of the key factors, that which are important when we select a polymer for delivery system. That is chemical structure and chemical composition. Of course, as I said, since it is a copolymer of lactide and glycolide, so their ratio, because lactide is more lipophilic, glycolide is more hydrophilic, so naturally the ratio will change the water uptake tendency and the subsequently erosion rate of the polymer. Yes, molecule weight, like any polymer, the higher will be, the longer will be the chain, more time it takes to degrade. So higher molecule weight, it will take more time to degrade, so it will give a longer drug release. Similarly, viscosity, what primarily generally we call for polymer, it is inherent viscosity. So naturally, higher the viscosity, it will take more time for polymer to degrade, and more or longer duration drug release can be designed based upon higher viscosity. And then becomes an important aspect is the presence of end group. Yes, like PLGA is an ester-based polymer, where but if you see, what about the end group? That the last monomer in the polymer chain, what is the chemistry of that end group, whether it is acid end group or whether it is ester end group, that also dictates that the nature of the polymer and how it will absorb the water and degrade. Morphology, yes, what is that, whether the polymer, the crystallinity of the polymer. So higher the crystallinity of the polymer, naturally more is the lattice energy, and it takes more time for polymer to degrade, means longer time of drug release can be planned based upon that. Monomer concentration in the polymer, when we can't talk about monomer, in what is the residual monomer of lactide, residual monomer of glycolide in the PLGA polymer, the microstructure, how the microstructure of the polymer, like lactide glycolide moieties are there, but what is the block length, whether it is a lactide glycolide lactide, LG LG LG, or it is a LLLGGG. So these are all the microstructure, etc. I will not go more into the details because of the limited time available, but these are some of the factors, and we will talk about some of these factors out of this in a little bit more elaboration in the coming slides. Before we move ahead, just very simply that talking about the, the molecule weight and inherent viscosity, as I said in the beginning, yes, for any polymer, these two are very important criteria. Where we say here viscosity means it is inherent viscosity, IV, what we call. So there is we know it's a direct, very positive correlation between molecular weight and inherent viscosity. Higher the molecular weight, higher the viscosity, means bigger is the molecule, more will be the molecular weight, more will be the inherent viscosity, and bigger is the molecule, more time it will take to degrade. It takes more time for chain to cut down to smaller chains and then ultimately degrade. So higher the molecular weight and higher the inherent viscosity, more time it will take for polymer to degrade, and it can give you longer duration of the release. That is why, relatively, if you want to have a 1 month depot injection, 3 month depot injection, so in 3 months, one approach can be you use a higher molecular weight, use a higher viscosity. Further, if you go on increasing the higher molecular weight and viscosity, you can even go for a 6 month delivery system. So this is just one example. And further, another important aspect is, the lactide glycolide ratio. How is the ratio of the lactide and glycolide? So that is, very important. We will talk more on that. But this is just, a brief about that, the degradation over a period of time. What we talk about the fraction remaining, suppose initially the polymer, and how the polymer erodes over time in terms of molecular weight, in terms of strength of the polymer, in terms of the mass. When we say mass, means it's weight, the weight of the polymer, which is injected into a body tissue, whether it is subcutaneous tissue or intramuscular tissue. So what is important to see here, actually, in the first glance, when actually the cleavage happens of the polymer chain, the ester bond cleaves, and the first of all is the molecular weight, which starts degrading. And subsequently, after the molecular degradation, the next degradation will be coming down will be on the strength of the polymer. Strength means strength of the particle, so its strength comes down, and lastly it is the weight of the polymer in the tissue which comes down. So most of the time for our study and comparison per se, normally for delivery based system, we talk more on the molecular weight. We go with the molecular weight. Strength becomes more important for orthopedic implants, where biodegradable orthopedic implants are there, where strength measurement becomes more important. But for our delivery system, generally, we focus on the degradation pattern of the molecular weight. And as we were talking about, one of the important aspect is, one of the important lever what we can use for selection of the polymer is that how much is the ratio of lactide and how much is the glycolide. As I talked earlier, lactide is more lipophilic in nature because one extra methyl group. So this is here, comes, suppose we have a polymer which is 100% lactic acid, means it has relatively more of a lipophilic component, that is lactide. So you can see here on this one, it takes more time, for degrade, means for a polymer to its half life, how many months it takes for the polymer to comes to its half of the weight, so here comes, it takes more time. So it means higher lactide means degradation will be slower. It takes more time to come to its half of the molecule weight or half of the weight, and naturally it means it will give you a longer duration delivery system if your lactic acid is high. So if you go on reducing the lactide, from 100% of lactide, suppose you come and releasing the lactide, you are releasing, decreasing the lactide, means 90 means lactide, means remaining 10 is glycolide. So you are reducing the lipophilicity and increasing the hydrophilicity. So your degradation kinetics goes higher, means your half-life of the polymer in terms of degradation is slowed down, and means you will get faster release from such delivery system made with this polymer. Further, if you go down further, like 75-25 means 75 lactide, 25 glycolide, further slow release in the sense means shorter duration of the release because half-life will come down. And we may think of, if we go on reducing the lactide, it will follow a linear pattern, but it is not so. Because what happens when you come here somewhere in 50-50 polymer, means 50 lactide, 50 glycolide, and if you go on increasing lactide more, when lactide becomes, say, more than 75 and glycolide becomes more than 75. Here if you see, lactide is 25, so lactide 25 means glycolide will be 75. So you may think, oh, that glycolide is more hydrophilic in nature, and if you're increasing glycolide, actually it should slow down further, but it doesn't happen so, because another factor comes into the picture, that is the crystallinity. Actually the glycolide, although it is it is hydrophilic in nature, but it imparts crystallinity to the polymer. And as we move on this particular side, that is the more and more glycolide, 25% lactide and 75% glycolide, and we go on increasing glycolide component on this side, since the crystallinity of the polymer as such increases, it because of the more lattice energy of the crystalline nature of the polymer, it takes more time for polymer to degrade. That is how, you see here his half-life is increasing again, and naturally with this, the delivery system, what it will be made, it will take more time relatively to it will take more time to degrade and longer duration. So if you want to make the fastest delivery system, say you want to release within a month, you have choice of going with 50-50, that is lactide, PLG 50-50 or so. If you want to make a 3-month delivery system, maybe you can use a 100% lactide. And similarly with high glycolide also will give you longer duration. So what I want to emphasize, with this, just a simple case study of LG ratio and how the parabolic nature of the influence, how the influence of this particular CMA of the polymer can be on the drug release of the finished product. Is it, my voice is clear? Should I go ahead? Right? Is it fine?
Yes, sir.
Yes, sir. Good. I will go ahead. Thanks. Yeah. So as we move ahead now, let us talk about in terms of that lactide glycolide of ratio, and just see that how much time it takes for its in terms of the weight. So the red curve depicts the polymer 50-50, where 50 lactide, 50 glycolide. So you see here relatively, it takes lesser time to degrade. So so that was, I was giving you example, if you want to make a 1-month delivery system, because it takes somewhere around maybe 35-40 days or so for the polymer weight, the weight to come down. So for a faster duration, say 1-month delivery system or so, you can use PLG 50-50. Similarly, if you see here, if instead of PLG 50-50, use 75-25, means 75 lactide, 25 glycolide. You see here, the residual weight, it takes more time to come down, means polymer degradation kinetics slows down, means you can, with this, you can think of having longer duration of the drug release. So this is the pattern of how the residual weight of the polymer comes down over a period of time when polymer is in contact with the aqueous fluids like biological fluids in muscle or subcutaneous tissue. And here you see that how actually the molecular weight of these two different monomers, these two different types of polymer with different monomer ratio. Again, the red line shows PLG 50-50, that is it takes again around 35 days by the time you do molecular weight to come down. And naturally if you increase the lactide ratio, means you increase the component of the lipophilicity, that is lactide, 75-25 higher the lactide, so it takes more time for polymer to degrade, means polymer kinetics is slowed down. And with this relatively, if you make delivery system, this will give you a slower rate, means higher duration of the release. This will give you relatively faster duration of the release. And this is a a very good case study in terms of depicting how different monomer ratio, if we change from 100% lactide to PLGA 87-13, means 87 lactide and 13 glycolide, and so on gradually. If you go on releasing the lactide component, and finally here comes 50-50. So, and this is the the study done when polymer was made using radioactive labeled C13 polymer, just to see that to monitor how is the radioactive remaining, which is showing that the degradation kinetics of the polymer. So you see here the 100% lactide, it takes more time, and remember this is in weeks, not in days. So it takes almost a year to degrade. That is how using 100% lactide, now we have delivery system in the market which release the drug for 6 months. You have Leuprolide depot injection available in the market, which where just one injection releases the drug over a period of 6 months. And if you go on increasing the glycolide component gradually, you see here how degradation kinetics is increasing. And if you come to PLGA 50-50, maybe it's almost within a couple of months the whole polymer degrades. So if you are making a 1 month depot injection, this may be a good choice of LG ratio. So yes, we talked a lot about lactide glycolide ratio, molecular weight, inherent viscosity. Another very important aspect is the acid end group. So when we talk, I think beginning, I just touched upon that. Yes, you have a polymer long chain, but what about that end chain? Where end group. So at the end group, you may be having an acid, for example, like it is a lactic acid, glycolic acid. So with lactic acid and glycolic acid, which is having hydroxyl group, which has carboxyl group, it will be ester polymer. But at the end, there may be choice. End group may be acid, or that acid end group you can esterify using some higher chain alcohol like stearyl alcohol, lauryl alcohol, and you make it ester end capped, what we call end chain. So this is just case study that if you take a polymer of a similar other CMA, when I say other CMAs, means in terms of the similar molecular weight, similar inherent viscosity, similar LG ratio, and simply you modulate with the end chemistry of the polymer. This is how it is. If you have an end group is acidic in nature, so that relatively it is wetted with water easily because it is hydrophilic in nature, being an acid end group. So what happened, this orange color curve, it is pertaining to acid end group, where being acid end group, it is more hydrophilic, it imbibes the water from the biological fluid faster. Since water uptake is high, what shows on y-axis is water uptake. High water uptake means water will go inside the polymer, it will degrade the ester bonds, and so on. The degradation will be faster. So for a faster release, if you use carboxyl end group polymer, relatively release will be faster. Similarly, if you use the same polymer, if the end group you esterify, let's say lauryl alcohol, you make it ester. So what happens, being an end group is lauryl ester, more lipophilic, it will prevent the water wetting of the polymer, mainly the water uptake of the polymer. So since water uptake will be lower, diffusion of the water molecule inside the polymer matrix will be slowed down. So you can imagine very well, it takes more time for water to go inside the polymer. It will take more time for the drug to get released. So, out of these two, if you choose the polymer with ester end group, keeping other parameters same, your release will be slower and you can make delivery system of longer duration, right? So these are, some of the factors which are there. So, now, just to summarize again, this slide I'm showing again, to you once again that the various CMAs of the polymer which I shared with you. As I told you, these are not complete. This is not the complete list, but some of the most important. So these are some of the levers which are in the hand of the formulation scientist, who can use these levers judiciously to decide the drug release kinetics and make a polymer delivery system. There are delivery systems in the market which are of 15 days duration, means you have to give every 15 days one injection to the patient. And on the other extreme, you have a polymer based delivery system which is one injection in 6 months. So all these parameters help you to design the release kinetics and duration of the administration accordingly. And as I said, that this polymer-based delivery system, I gave you initially examples of leuprolide and example of decapeptyl, which were the initial one. So at this moment, these are, some sort of a dozen delivery systems which are available in the market, which are depot injection. And most of them are basically PLGA-based, polyester-based drug delivery systems. Right? So initially, I mentioned one share of Munir Niazi, but that is just to highlight that it requires a serious efforts. If the generic industry has not yet got the success for making a generic of such polymer based delivery system, I'm not discouraging that we can't make it. We can make it, but it requires perseverance, it requires efforts, it requires a lot of hard work also. So I said, one share, and I will end also with one more, share of Aslam Beg. What Aslam Beg has written, that उस बेवफा की आंख से भी आंसू निकल पड़े, हसरत भरी निगाह बड़ा काम कर गई. So what is important is, hasad bhari nigah means perseverance, means the effort, means the continuous hard work also. And that is what my message to the students here, that we should not get deferred with the failures sometimes which may come because of when we work upon the complex delivery systems. Failures will come, but we should work with hard work, perseverance, passion, and there is a lot more work to do, lot more delivery systems to crack, so that we can have these delivery systems available for the patients worldwide. So with this, I will just say thank you, and please feel free to put any questions, if any. Thank you.
Thank you, sir. Definitely, sir has given a wonderful talk about the PLGA, which is a US FDA approved polymer. And even I am also working over this polymer for antigen delivery. And sir has summarized that the several factors like molecular weight, viscosity, presence of end group, which is a functional group, morphology affect the drug delivery using this polymer. And sir has given also about a wonderful words about the success story of several depot injections. So I request the audience for any question from the sir.
Thank you, sir. So now I would like to invite Majumdar sir to brief the speaker.
Thank you so much, sir. Now I would like to have Dr. Anupama ma'am to felicitate with certificate of excellence to Deepak ma'am.
Dheeraj, words are less. Only thing is that एक simple thank you से बोलूंगी for you accepting our request, and it's so wonderful. But my faculty wants to say something to you. Can you hear us?
Yeah, I'm listening, sure. Yeah.
Yeah, sir, this one is for you. कटना पाई किसी से चाल मेरी, लोग देने लगे मिसाल मेरी, और मेरे जुगलों से काम लेते हैं जिनसे बंद है वो चाल मेरी.
Thanks a lot. So nice.
Thank you. We will be stopping the screen and coming over to the certificates. Okay, give me a minute. Yeah, and as just a gesture, please accept our. We will be sending it by email to you, and as Sir has rightly said, Dr. Majumdar, that we are looking forward for a day when you come to the ASU campus. खींच हम आपको किसी तरह कभी तो ठीक है धीरज.
Yeah, sure.
Okay, thank you.
Definitely. Thank you. Thanks to all for kind patience and listening. Thank you very much.
Thank you, sir.
Now, I would like to invite Professor Bansini Malhotra, DST-SERB Board, Government of India Distinguished Fellow and Technological University, Delhi. Over to you, sir.
Now I would like to call Madan sir.
Okay. Thank you. Sir, Professor B.D. Malhotra received his PhD from University of Delhi in 1980. He has published 329 papers in high impact factor journals with 23 113 citations, and in addition with the 82 H index. And Sir has also published 12 patents in India and overseas, and co-authored textbooks on nanomaterials for biosensors, fundamentals and applications. He is a recipient of the National Research Development Corporation Award 2005 for invention on blood glucose biochemical analyzer, and is a fellow of the Indian National Science Academy, the National Academy of Science, India, and an academician of the Asia Pacific Academy of Materials. His current research activities include biosensors, nano biomaterials, biofuel cells, ordered molecular assemblies, and many more. Dr. Malhotra is currently a DST-SERB Distinguished Fellow and an adjunct professor with the Development of Biotechnology, Delhi Technological University of Delhi. Now I request Sir to please start their talk.
Thank you, sir, for the nice introduction. And at the outset, let me thank the organizers of the workshop, national seminar, in particular, Professor Majumdar, Professor Diwan, and Dr. Rani, for the opportunity given to me to talk to you, to be with you this afternoon, and tell you about some of the things we have been trying to do, or I have been trying to do with other colleagues, colleagues to work on biomolecular electronic devices. At the outset, I should also mention that we are writing a book which is just completed on nanomaterials or graphene-based biomolecular electronic devices, and which will be released by elsewhere sometimes in about few months. So at the moment, I decided to talk on how nanomaterials based conducting paper can be helpful towards the development of biomolecular electronic devices, in particular biosensors for cancer detection. Well, this is, I don't know why is it not moving. I can't move it. I can't move the slides. Can I do it again?
So please share your screen.
Yeah, I have to share the screen once again. I can't see that where. Can you, where is my screen? Share the screen.
Sir, this option is there to share screen. So
Where is the option? I have just shared.
आप एक बार PPT ओपन करिए सर और ये ग्रीन वाला जो शेयर स्क्रीन वाला बटन है जो आपने अभी स्क्रीन शेयर की थी वैसे ही करिएगा सर एक बार
स्क्रीन शेयर तो करा, अभी उसके बाद जा नहीं रहा आगे।
सर एक बार PPT को ओपन करिए।
Shall I start it again?
एक बार।
Yeah. Oh, where do I go?
सर अभी जैसे आपने किया था it was visible to
Yeah, I'm opening it. Yeah, how shall I pump it? Shall I double click this?
Yes sir, double click on the green button नीचे जो है share screen. Yes sir.
Can you see my slide?
Yes sir, it is visible. Yes sir.
Okay, so this is where I work, which is called Delhi Technology University, and it's a renowned institute, and it's a pleasure to work at this prestigious institute. What I'm going to do is to tell you why do we need, what is a biosensor and why do we need biosensors or a bioelectronic device. And as I said in my talk, in my title of my talk is paper. Why do we, what is paper and why do we want to use paper to fabricate biomolecular electronic device? And then can we make it electrically conducting? And the conducting paper can be used to investigate cancer, and in this particular case, I have decided to talk on a cancer, detection of a cancer biomarker called carcinoembryonic antigen. And there are large number of the cancer biomarkers, which you will see in a few minutes. So, and I also decided to talk, take a few studies, depending on the time, of course, on how the conducting paper-based devices can be used to detect carcinoembryonic antigen, which is essentially a cancer biomarker. So biosensor is an electronic device or is an electrical device that converts a biological response into a measurable signal via a transducer, and the transducer can be electrical, piezoelectric, optical, and or even electrochemical transducer. So, and it essentially makes use of certain receptors, and receptors can be an enzyme, can be a deoxynucleic acid or peptide nucleic acid or aptamer or antibodies or living cell. This morning I had a chance to talk to some young students for the upgradation of senior research junior research fellowship to senior research fellowship. So I asked the person concerned is that why is it that we can't have room temperature antibodies or monoclonal antibodies which can be useful or which can be used to construct a biomolecular electronic devices. So these are some of the analytes which are shown here. Analyte can be a glucose or a urea, glucose, urea, or cholesterol or lactate. Somebody was mentioning a little while ago, and we have had the opportunity to work on a variety of biosensors which can be used to detect the desired analytes. And the receptor, of course, can be DNA, even antibodies and whatever, and the transducers can be electrochemical, optical, or piezoelectric. And what really happens is, as a result of the interaction of the receptor with the desired analyte, you have a certain output, and that output can be electrical or optical or piezoelectric, as I mentioned a little while ago. But the trouble is that most of the receptors that we are carrying in our bioreactors, is they require certain conditions to get the maximum output or perform the best. The condition can be pH, temperature, or even viscosity at times. The desired biosensor should meet the desired assured guidelines given by the World Health Organization, and that's one of the best things is that it should be sensitive, should be specific, and the data that you get from a particular clinic should be matched with the ones which are available from other clinics, or even maybe perhaps stationed at the moon at the moon. So it should be affordable, it should be user friendly, it should provide rapid results, not like the ones which we used to be in earlier days. You go to a clinic or biochemical laboratory, and they will take your blood and make it into a serum, and then of course you get your results. And these days the results are flashed on your smartphone, but earlier you had to go and personally collect it. So in other words, we want the data like glucose test strips in a few seconds, and we want the reliable data, and we want cost-effective data. And then the user should not be asked, not have to do anything with the system or with the device. What the user wants is the accurate data at the affordable price. So more about these information about what a biosensor is. There is a review article which became only this month, about a week ago, on encodation chemistry reviews, which has a high impact factor. So a biosensor should be low assay time, should utilize low sample volume, and it should be capable of real-time data. I want glucose data of my glucose going into my system as a function of time as such, so that I know what to do next, or what to do next in the next second, as far as my body is concerned or living is concerned. It should be cost effective, it should be user friendly, and it should be environment friendly. Those biosensors have large number of applications. You can use that biosensor for environmental monitoring. You can use it for pollution monitoring. You can use it for food toxins. That is where pharmaceutical industry comes into play more efficient and more rigorously, actually. And in fact, the fruit that we take consume, we should also know as to when to pluck the fruit from the tree, so that all the desired ingredients that are carried by the fruit, they don't get destroyed, or they don't get over-ripened or destroyed. That's a better word. So, and we need biosensors not only in vitro but also in vivo, and that is where biocompatibility will come into play. And then what essentially I'm looking for is not only the biomedical devices that I can implant into my system, but also I need bedside glucose monitoring, or home glucose monitor, or home cholesterol monitor, or home urea monitor as such. So this is what it is, and we produce the glucose biosensor while I was doing my stint at the National Physical Laboratory of the country, which is stationed in Delhi. And I worked for around 35 years there. In fact, received my PhD from Delhi University while working at National Physical Laboratory. So glucose, I don't know if you can see it. It's a test strip basically, which consists of, it's a miniaturized electrode. Let me see if I can start the, well, never mind. I will have to switch it off to show you what I am holding in my hand. But nevertheless, it's a miniaturized device which consists of a anode and a cathode and also a reference electrode in silver chloride. So, but the glucose test tube that you get in the market, they are basically two electrode devices. One is working electrode, which essentially consists of silver, which consists of graphite, and then it has a, underneath the anode, it has a silver layer, so that the whatever reaction goes on to the miniaturized electrode, miniaturized area in here, in this area, when you cover this area with your test specimen, it can be blood, can be sweat, it can be even saliva. And then the reference electrode in this particular case is silver chloride. So when you cover this area with your test specimen, there is a biochemical reaction that occurs, and that biochemical reaction produces certain current, and that current can be monitored as a function of the glucose concentration contained in the test specimen, which can be serum, which can be blood, and whatever. So if more of you who are interested, there's a company which is based at Bangalore called Pulsatum Healthcare Private Limited. If you put Pulsatum Healthcare Private Limited in Google, Baba, you will get to know the price. And the price of this particular glucose test is around 10 rupees per test, 10 rupees per test. And then how it works is you have glucose, which you want to monitor, and glucose oxidase is an enzyme which is injected into the test specimen, which is immobilized onto graphite, and the glucose content and the reaction of interaction of glucose with glucose oxidase, which is a protein or an enzyme, it releases certain product, and glucose oxidase gets reduced, and the product is gluconolactone and hydrogen peroxide. And this glucose oxidase reduced, which is reduced, and we make it react with a mediator, which can be organometallic called ferrocene carboxylic acid, or even ferrocene will work. So as a result of the interaction, mediator gets reduced, and the glucose oxidase gets oxidized, and the reduced mediator, we switch it on, we apply a certain potential around 0.6, 0.4 volts, which will transform the mediator or reduced mediator into oxidized mediator, and in turn also release 2 electrons. So number of electrons generated will be dependent on how much glucose is there. So the glucose can be monitored, and this is what is available in the market today. Lots of tests are available. Lots of companies are selling this product, and one of the companies, as I said, we gave it to 3 companies, and only one of the companies succeeded. And so this is what I had shown you, what the characteristics of a biosensor is. And the first example I give is of a conducting paper-based biosensor and a made of a cellulose. Cellulose paper consists of a cellulose. It's a natural paper polymer comprising of a linear chain of beta 1 to 4 linked glucose, and this paper is being manufactured or can be fabricated by debutering a dilute suspension of cellulose fibers via the filtration process that is followed eventually by pressing and heating. So the mechanical properties of the paper are because of the microfibular structure and resulting due to hydrogen bonding, right? So paper has many advantages. It's flexible, it is easily available, you can destroy it easily, it's biodegradable, it is cost effective. And then you can make it electrically conducting by either coating it with a metal called gold, silver, or even metal iron oxide, or molybdenum oxide. And then you can also, as I will show in the later, you can also use nanomaterial like carbon nanotube, or even a conductive polymer, which can, or carbon nanotubes, polymer nanocomposites, to make it electrically conducting. Why do you need it electrically conducting? I will show you in a minute. So this is it, and conductive paper has several applications and large number of You can use it as a field effect transistor or organic field effect transistor. You can use it as in a battery, capacitor, nano generator, biofuel cells. We have recently developed interest in biofuel cells or biofuel batteries. And as I already mentioned earlier, you can also implant carbon nanotubes into a conductive paper to make it electrically conducting. And one of my former colleagues who is now works with Naipur Guwahati, he worked on how to utilize a conductive paper to get troponin, which is a hard biomarker. That is, the concentration of troponin beyond a certain limit can tell you whether you are going to get a heart stroke or not. So this is it, and then I mentioned about one paper, and then if you are more interested in trying to get at what a flexible conductive polymer or conductive paper can do, there is a recent review article we wrote in additive manufacturing, which can be utilized to, which essentially contains the information on how flexible devices or electronic devices can be made. So I am interested in using biosensor for cancer detection, and cancer is abnormal and uncontrolled cell growth, and that can occur due to accumulation of specific genetic and epigenetic defects that are can be both hereditary and environmental of origin. And then unregulated cell growth leads to the formation of tumor mass that may become independent of normal homeostatic checks with time. And tumor cells are resistant to apoptosis, so that is where the cancer gets a lot, and they kill or they retard the anti-growth defenses within the body. So you are all most of you are on biology, so you know how a cancer originates, and then this is how the apoptosis occurs, and the necrosis can results into the origin of cancerous cells. So cancer can be detected using biopsy, which is the gold standard. You can use computer tomography, you can use molecular techniques like polymer chain reactors or cytopathology, and the disadvantage of that, none of us can operate it ourselves, and you have to seek test assistance or information, or you have to take help of a professional. And the results may not be easily quickly available. It may take a couple of weeks, and the cost of operation is high. So, and moreover, it is not accessible into remote areas. So cancer biomarkers is a molecule which is created by a tumor or a specific to the presence of cancer. And there are more than 200 or maybe 300 cancer biomarkers now. And then the cancer, if you know the concentration of a biomarker cancer, all of us would like to know whether my system that I am carrying is likely to develop some disease or more specifically cancerous disease. And if yes, what type of cancer it is it and what is the optimal drug for my cancer? And then pharmacodynamics is also very important, we would like to know what is the optimal dose for my body to cure a cancer. And then I'm more interested in trying to find out whether the cancer will return. A large number of cancer biomarkers are available, or cancer disease biomarkers are available, and I chose to work on only carcinoma embryonic antigens or CEA. And a person who is now in Naipur, Guwahati, he worked actually on this particular area for his PhD at Delhi Technological University. So CEA is a glycoprotein which has a molecular weight of 180 kilodalton. It's a tumor biomarker, and its concentration can tell us, or presence or concentration beyond a certain range will tell us whether we can have it will shortlist the cancers. Actually, it can be colon, it can be lung, it can be ovarian, or it can be breast cancer. And then detection will be helpful in therapeutics. And then CA serum level can be correlated with the depth of tumor invasion, and normal CA level is at less than 5 nanograms per milliliter. If its concentration goes to beyond 5 or even maybe even 20 nanograms per milliliter, so we are sure to one is sure to have one of the cancers listed, which I listed just now. So polyaniline is a conductive polymer, very widely known, and we did some work while being at Delhi Technological University. It has a solution processable. It's a large surface area, and it's a good electrical conductivity. Presence of amine groups enables conjugation and facilitates enhanced electrochemical properties. So this is how it was done, and antibodies of CA were bought from the market, and they were coupled to polyaniline after onto a paper which was modified by gold. Gold was sputtered onto conducting a paper that I mentioned, and then we deposited polyaniline onto it, and then antibodies of CA were immobilized. And then after we fabricated this particular electrode, we put BSA to we immobilized anti-CA through EDC-NHS coupling, and then BSA was done to nullify the unfilled sites or vacant sites, which were not. If we do not treat it with BSA, they will contribute to this current generated due to this. And then, of course, we want CA to be determined. CA is an antigen, and we have CA antibodies of CA immobilized onto it. So as a result of the interaction, you have the you can monitor, you can get the current or the change in optical any property that you are interested in. So these are some of the electrochemical deposition of polyaniline, and these are the characterized using scanning electron microscopy and X-ray diffraction. And what you see here is how, as you increase the cyclic voltammegram basically, and you can see that as the thickness increases, current also increases. And then the SEM was taken to find out whether, so the gold was sputtered, metal gold was sputtered onto paper to make it electrically conducting, and the polyaniline was eventually deposited onto it. And then we found that this particular electrode or this particular sensor has a sensitivity of 13.9 microamperes nanograms per milliliter per centimeter scale. And we also tested this electrode with the results obtained from the hospitals, and the detection range for this particular biosensor was 2 to 20 nanometers. So the specimens were also taken obtained from Rajiv Gandhi Cancer Hospital, and it was found that this particular system was accurate up to 5%, and then the response decreased, of course, to about 73% in about 22 days. So in up to 23 weeks, you could utilize it. So paper use, I didn't mention it earlier, it was Wadman paper 1, and gold was sputtered, and then the problem was that I wanted sensitivity is still low, it's in microamperes, and the cost of the gold is also pretty high. So we use conducting polymer, nanomaterial, reduce graphene. You all know it was it was awarded Nobel Prize somewhere in 2009, I think, 2009, and then oxidized form of graphene is electrically insulator, and if you reduce it, it becomes electrically conducting. And we modified the conducting paper by reduced graphene oxide and, of course, for CO detection. And why do we want to reduce graphene? It has a nice, desirable electrochemical properties, and functional groups enable it to couple the antibodies of CA onto it conveniently, and then the charge transfer properties are also very high. So what we did was P dot is a conducting polymer which is available, which is water soluble, and polystyrene sulfonate is another ionomer, and we made it, and the PSS is negatively charged, but the P dot is poorly soluble and it's positively charged. So that is why they are utilized. And P dot PSS is a conductive polymer which has a high to surface volume ratio. It's a thermally stable and it's a better orientation, and it has a good electrical conductivity. It's very widely used. So this was a schematic of what was done in here. And what we found is that 3% of ethylene glycol solution is used to dip, is used to, what I meant to say is the paper is put into a solution which contains 3% ethylene glycol. And then onto it we put reduced graphene oxide, and after the surface is prepared, the immobilized antibody was CEA or carcinombrenic antigen, and then this whole electrode is treated with a test specimen which may or may not contain CEA in the desired range. So we mentioned we did the electron microscopy investigations and also the transmission electron microscopy investigations, and we found that this particular electrical conductivity of this particular electrode goes down, decreases up to 20% after about after it is molded by 30 times, and then the reduction in the conductive conductivity is due to tearing of cellulose threads itself. So what I'm trying to say is that what I show you in this particular video is that the conductive paper that we produce using RGO, when you connect it together, the LED will get lighted up. Just a few seconds, and then when you it was so that means the paper is electrically conducting. As a non-electrical conducting paper will not produce you any light or not switch on the LED. So because the time is limited, I am so I will go to the next slide. So the mechanism of the electrical conductivity is that there is expulsion of PSS molecules out of the systems as a result of the treatment of this particular paper with the 3% ethylene glycol. So you can see that when you treat it with the ethylene glycol, the electrical conductivity is raised by at least two times at least. So which is a good result. And yeah, and then the experiments were also for X-ray photoelectron spectroscopy was found out as to what is the origin of electrical conductivity in this particular system. And then we found that it is as a result of the treatment of the electrode with ethylene glycol, there is a removal of polystyrene sulfonate ions from the film itself. And then the linear range of this particular electrode was found to be 2 to 8 nanograms per milliliter, and the sensitivity is about 25.8 microamperes per nanogram. So which is good, and stability is also 21 days. But what is not good was that the cost is low, of course, and then we want a better detection range is only 2 to 8 nanograms per milliliter, so which motivated us to go to another work which deals with carbon nanotubes, modified conducting paper, and everything else was the same. We did use ethylene glycol. Maybe the percentages of the ethylene glycol treatment was around 5% in this case. Earlier it was 3%. So why do we want to use carbon nanotubes? There was a Nobel Prize also given for this, which had a good conductivity and enhanced electrical conductivity, and it can result into high surface area. And then we used, instead of we use also formic acid, which is a high dielectric constant material. And then after having treated with, we of course you put carbon nanotubes into this particular system to make a composite and to make it electrically conducting as such. So the whole thing was dried at 100 degrees centigrade in an oven, and after which formic acid treatment was given. So these are the results of the X-ray diffraction investigations. Raman spectroscopy, carbon nanotubes can be viewed through Raman spectra generated onto this particular system, and we found that the presence of 1361 centimeter wave numbers and 1562, and the shift of this to 1570 centimeter was indicates the existence of interaction between carbon nanotubes and the conjugate paper. So you can see that the conductivity has got improved as a result of the treatment with carbon nanotubes and also formic acid. And then this particular system was also used to get the to determine the CA concentration as such. And these are some of the SEM images and some data as to how the electron transfer rate changes as a result of its treatment with formic acid. So stability of this particular is low, about 18 days. We want to use the electrode for as long as we live. This is a weakness. Linearity is 2 to 15 nanograms per milliliter, and the range is about 1 to 100 nanometer. So out of these three systems that I mentioned, you can see that the cost is not is low in on all the three systems that I described. But then the limitation is the sensitivity is low and the sensor doesn't cover the required physiological range. And then another work which was done go on to conducting polymer fiber of polyethylene PBA, polyvinyl alcohol actually. And then what we did was we made the paper conducting. How much? Yes, you have any question? Please, I have limited time. Am I okay? I'm knowing you're okay with the time, please. Nobody says. Anyhow, I'll try to finish it as soon as possible. And then this conducting paper was made by using the polymer solution of polyvinyl alcohol, and then we found that this particular system, everything else was the same, but only conducting paper and P dot, as we found that the conducting paper P dot PSS tried electrospun nanofiber along with conducting paper like P dot SSS gave us the better tensile strength, or it was more rigid. And we made some contact angle measurements, electron microwave investigations, and we found that this particular system has, at the moment, it is probably one of the best, if not the best as such, because the next system that I'm going to show you, probably with iron oxide nanoparticles, has a better result. Iron oxide nanoparticles are important, not only because of that they are biocompatible, but then they are, as I will show in a minute, as to they are not so expensive either. So stability is 22 days, after which the current decreased by 21%. And now the most important point was that when we burn it and then do the element analysis, we didn't find any analysis. So there is an environmental safeguard towards it, to this electrode. So you don't have to worry about the environmental hazard as such. So as I mentioned, there were no carbon, oxygen, sulfur, potassium, and even calcium when the ash from the ash which was obtained after burning, of course. So this particular system has 22 days, and there's a good linearity from 0.2 to 25 nanograms per milliliter. And this paper has got attention of a lot of workers. I promised about these iron oxide nanoparticles, they included P.SS, everything else was same, and we did the we made the connecting paper by using iron oxide, and DMSO was used as the solvent to, in fact, iron oxide nanoparticles were, along with P dot SS, were resolved into DMSA, and then antibodies of CA were put into by electrophoretic process as such onto it, and BSA was used in all the cases that I described. BSA has been used in all the cases to passivate the unfilled sites or the vacant sites that may emerge. So what I'm essentially trying to emphasize is that this particular electrode has a sensitivity from 2 to 100 nanograms per milliliter, and of course, out of which linearity is only from 4 to 25 nanograms per liter. Sensitivity is around 10.2 microamperes. Microamperes per nanograms per milliliter per centimeter square. So, and the electrodes were also tested with the test specimen obtained from hospitals, and the result, if you see it, this particular system perhaps is doing the best, around 34 days, and it has low viscosity, low cost, and then its sensitivity is also very high. So we have used polyaniline, reduced graphene oxide, carbon nanotubes, iron oxide, nanoparticles, and the efforts should be made to improve the stability and the detection range. And we also would like to see whether some of the electrodes paper electrodes that I mentioned can be used to detect coronavirus. So we have written a review article onto this, and some of you are in fact, this review article is having a lot of potential, a lot of demand, and a lot of people are following the work after reading this. So these are the electrodes that I described. They have implications in flexible electronics, thermodynamics, environmental monitoring, food safety, macrofluidics, and we also have done one work on paper biofuel cells as such, and that paper is published in Biosensor Bioelectronics. And aim is eventually, can I have a transparent paper as such, made of the summer system that I described. So there's a lot to be done, and work that I described was of Dr. Saurabh Kumar, who is currently in Naipur, Guwahati, who just joined about a few months ago, and Dr. Sabeen Kumar is a inspired faculty at automotive chemistry. And Dr. Saurabh Srivastava also has a job now in Banaras. Magnus Wilander is from Sweden. Shayan Augustine now not a PhD scholar anymore. She's a postdoc now in Korea. Anita Das also works in New Zealand somewhere. Mohammad Amar is doing some business somewhere. Virendra Kumar Yadav is a biobank manager in the Institute of Liver and Biliary Diseases. Dr. D.C. Dawal is for Rajiv Gandhi Cancer Hospital, and Niharika Gupta, of course, is currently working on transparent conducting polymer device. So some of the colleagues I am grateful to who made this possible for me to talk to you, to be with you. Dr. Chandramouli Pandey, Dr. Saveen Kumar, Dr. Shayan Augustine, Niharika Gupta, and Dr. Saurabh Kumar, Dr. Saurabh Srivastava. They are the ones who have in fact contributed towards this. Some are with me, some are in USA, some are in IIT Delhi, and some are from IIT Delhi. Thank you for your time, for giving me the time. If there are any questions, I will be glad to address it. Thank you. Life is an experiment. The more experiments you make, the better it is. Thank you. Any questions, please?
Thank you, sir. Thank you, Professor B.D. Malhotra, and given a wonderful talk about the nanomaterial-based biosensors. Definitely, research on medical devices is the need of the time, and you have talked about that nanomaterial-based biosensors have been predicted to significantly contribute to emergence of new molecular diagnostic techniques for patients suffering due to cancer. And a major problem with preventing faster development biosensor relates to the fact that cancer is highly complex disease and it is highly prevalent mainly in the northeast region of the India. And the oncologists heavily rely on the histopathological characterization using a few biomarkers of tumors. Some of the biomarkers include changes in the gene, as you are told, gene expression, epigenetic and genetic signatures, protein profiles, and post-translational modifications of proteins. These molecular signatures offer new opportunities for the development of biosensors for cancer detection, as you told. Besides this, these interesting bioelectron devices have been implication in drug delivery. Now I request the audience for any question from Professor B.D. Malhotra.
Thank you, sir. Questions can never be foolish. Answers can be different. You please ask any question. Hello. Yes, please. I can't hear.
Thank you so much. Thank you so much, sir. So now I want to invite Professor D.K. Majumdar, sir, to brief the speaker.
Hello. Thank you, Professor Malhotra, for
Any question, any query?
It would have been better had it been in offline mode, but we can't help this corona time. So I would expect, say, in coming days, they physically come to our campus and expose our students to these new aspects of management. Thank you once again.
Thank you so much. Thank you.
Thank you so much, sir. Sir, now I would like to have Dr. Majumdar, sir, to felicitate Certificate of Excellence to speak more and chairperson. Yes.
I would like to thank the chairperson from NIPAR and Professor Malhotra for sparing their valuable time in giving us this beautiful lecture and moderating this thing. Thank you.
Thank you, sir. Thank you.
Thank you, sir. Thank you.
Thank you so much, sir.
Thank you.
Thank you so much, sir. Now let's have a second chair, Miss Navita Srenan. Navita ma'am is pharmacy graduate with an MBA in marketing and has 20+ years of experience in operation, marketing, leading people, teams, and complete business units. So she founded and built a world-class team at CRO and has held various leadership positions at various pharmaceutical and services organizations. Our stories. Her role involves ensuring we have the highest level of satisfaction and engagement with our stakeholders by effectively nurturing and leveraging our human and technical capitals at all the times. Now, I would also invite co-chair Professor D.K. Majumdar, sir. Now, I would like to invite.
Now I would like to invite Mr. Indu Bhushan. He's Chief Executive Officer and Director, Sterlite India Private Limited, Bangalore. He's graduated in pharmacy from Birla Institute of Technology, Ranchi in 1987 and post-graduation from Punjab University, Chandigarh in 1989. He has been associated with development of pharmaceutical products in various capacities during last 3 decades. In the past, he played key role in setting up the facilities and resources for the development of generic and specialty pharmaceuticals in leading Indian multinational companies. He is the co-author or co-inventor of more than 50 patents or publications related to pharmaceutical products, composition, and processes.
Now I would like to call D.K. Majumdar sir to greet our guest with green certificate.
Okay. Bhushan, I would like to welcome you to APJ University for your talk, and I would like to accept our e-certificate, green certificate. We'll plant a small sapling here in your name, and we offer this green certificate. Kindly accept it.
Thank you so much, Professor Majumdar.
Thank you so much, sir. Thank you so much. Now I request Mr. Indubhushan sir to present his topic on hot melt extrusion, pharmaceutical product development and manufacture.
Are you able to see my screen?
So 1 minute. Yes, sir, you can share now.
Are you able to see it now?
No, sir. Click on sir that plus green slideshow button to share the screen. On Moodle itself, on Zoom itself, there is one green color plus sign slideshow.
Okay.
So yes, from that begin. सर जूम पे ही ना नीचे एक ग्रीन कलर का बॉक्स बना होगा तो उस पर क्लिक करके आप एक बार कर लीजिए वो उसके स्क्रीन शेयर हो जाएगा
Share screen and then what to do?
So wait, we are finding the video. Sir, if you are not able to share, I'll share your video.
Yeah, please share from your side. I will talk and then I will ask you to change page whenever required.
Yes.
Sir, is it visible to you?
Yes, yes, thank you so much. Let me start with thanking APJ Satya University for inviting me and allowing me to present a technology which was developed during World War II, however, did not get the recognition which it deserves. Um, it has been used extensively in pharmaceutical world, particularly in solubility enhancement. But if you look at number of products which got developed using hot melt extrusion technology, those can be counted on fingertips. So while polymer and plastic industries, they have utilized a lot this technology, pharmaceutical industry somehow struggled. There are many advancements which happened in last 20 years, which makes it now much more versatile, much more useful, which I am going to cover during the application part. So if you look at today, hot melt extrusion technology is primarily used in solid oral and transdermal formulation. In few products, if you see, it is used even for making implants. So those are the application area which needs careful understanding of many criticalities of hot melt extrusion, because on one hand, a pharmacist want a product with certain attribute. On the other side, it involves knowledge of highly diversified field. Go to next page, please. So when we talk about criticalities associated with hot melt extrusion, remember that it involves engineering sciences, because the process design is something which is very close to engineers, not pharmacists. On the other side, what product we make out of hot melt extrusion, they are primarily to modify PK or PD profile of the product. So therefore, a understanding of PK/PD is also required. Of course, it is dependent on the composition. A lot of polymer goes in hot melt extrusion. So a knowledge of polymer rheology and other pharmaceutical aid is also essential to design a process of hot melt technology and running it successfully at commercial scale. Next slide, please. So when we talk about the requirements in terms of process and application, what we see that the main area of application in solid oral is dissolution enhancement or dissolution modification. Traditionally, it has been used to make a solid dispersion or solid solution of water-insoluble drug with water-soluble polymer. That was the traditional use. And in recent time, we have seen controlled release product or even delayed release product have been formulated using this process. In few cases, in our lab, we could mask beta test of the product using hot melt processes. And of course, because of physicochemical changes, which is so significant during the process, we are also talking about stability of API or the finished product. So essentially, this process involves compounding or fusing, which is typical word used in plastic industry. Pharmaceutical industry call it as melt fusion, which involves structural modification, because we are talking about a physical change and a chemical change as well, which results in amorphous or solid solution many times. Many times what we see that if the drug is not forming a solution in the polymer, then we see micronization or nanonization. So particle size reduction to micron, and in few cases even to nano size, we have observed in the process. Next slide, please. So when we talk about hot melt extrusion, there are many variants currently available. Most of the people are familiar with the single screw extruder or counter rotating twin screw. I'm here to discuss more about co-rotating twin screw because it offers certain unique advantages and which makes it much more suitable for pharmaceutical application. The first thing is ability to run clean as a result of self-wiping profile. So in co-rotating, remember both the screws are moving in one direction. It may be clockwise or counterclockwise, but both the screws move in the same direction. What it results is a self-wiping ability which provides mixing as well as conveying work. And these are very essential during the extrusion process, particularly when the product is or the material is processed at very high temperature. Apart from that, different types of geometry of the screw can be arranged in order to do various types of the work, which we would like to see in the next slide. Um Please move to the next slide, please. So when we talk about co-rotating twin screw, we are talking about many different kinds of action or activity. When we see from outside, it looks one equipment. But remember, depending on the screw type, depending on the screw configuration, we can carry many different types of work which are essential for producing different types of attribute. Some of them are as follows. For example, shearing effect. We all know that shearing is primarily mechanical in nature. Typically, low shear rates produces heating or melting of the polymer or the drug substance. Whereas when it is more than desirable, it can cause breakdown of polymer or even the drug. That's something undesirable. So we have to always balance in the process what is desirable, both in terms of quality and quantity, and what is not desirable. Until unless we know those limits, we are not able to run it successfully for a very long time. The other type of work which is or activity which happens in the processor is kneading. And we are all familiar with kneading operation because we very commonly follow this in our kitchen. And kneading means elongation or folding or twisting or even compressing. Here again, while this is desirable for certain activities, if we do it very fast or very high pressure, then it may break the particles. So we have to be careful when we talk about the intensity or the amount of work which is being carried out in the processor. Last but not the least is stirring or churning rate. As I said earlier, this processor or this process is very good mixer. So we need to mix a lot of material and that too highly viscous in nature in the processor. Therefore, an efficient churning rate is highly desirable. However, when we see that sometime it may lead to again breakdown or erosion of the particle, and that is what is responsible for many times micronization or nanosization. So let us see what shearing does. Shearing helps in dispersing agglomerates or cluster of particle into smaller size in the matrix. Uh, Next, please. Kneading on the other side, helps in bringing wetting, because here in the process, we do not use any solvent. So hot melt extrusion process is typically without any solvent. And without any solvent, what we would like to accomplish is wetting. And that happens because we increase molecular mobility of the polymer and the drug both by heating and mechanical energy. That helps in wetting. But to do that, we need kneading work, which helps in wetting. One step ahead, please. In terms of stirring, one more forward. One step forward. Okay. So when we talk about churning or stirring, that helps in distribution. So we see the entire mixing as a two different type of work. One is dispersion, which basically helps material getting broken into smaller pieces. And distributing is homogenization, where these broken particles are distributed uniformly in the entire matrix. Next slide, please. When we look at the processing zone, so you would see the entire machine, but here what we call it as extruder processing zone, it has multiple zones. So it will look like a barrel. However, inside, if you look at, it can be divided into many zones. For example, conveying pump zone or action vessel zone. Action vessels zone are those where a specific type of work is being carried out, whereas in conveying pump zone, the material is typically conveyed with surface renewal, and we can even think of applying vacuum from outside, or we can think of adding liquid from outside in those zones, in conveying pump zones. So in total, what you see here is a processor which has ability to add liquid and even gases from outside, while the solid melts through the screw, and a mixture is made which is taken out as a hot molten very viscous liquid. Next page, please. Yeah. This page talks about degree of fill, which is yet another critical factor inside the processor. So when we talk about various geometry or various types of screw, basically what it does is it decides type of work, type of not only work, but how the material is going to flow inside the processor. The picture indicates that when the material enters in the processor, the material does not occupy a screw completely, because typically what we keep is a conveying element initially, followed by what we do is we would like to knead it before we start melting it. So those screws which are covered completely with solid, they are designed or they are kept with a certain purpose of activity such as wet kneading, or later on a milling or a melting operation. In the end, you will see that once the material is molten as viscous liquid, it is required to be pushed out through the die, and therefore what we call it as metering zone has a very small pitch element which produces a very high pressure, and which helps in extruding molten mass through a smaller aperture die. Now, once the material is molten and it is extruded, you can either give a particular shape or size, or cut it as pellet, or even you can think of making flakes after cooling it. The entire purpose of extruder then becomes doing a phase transition of two solid, making them miscible or wet with each other. Either you can think of making a nano or micro size particles, or even a complete solution which gives amorphous solution at the end. Next slide, please. I would like to explain what happens in milk extrusion in terms of wetting. As I told, there is no solvent which is used here. So we need to carry out wetting by the shear energy, which is typically mechanical energy. So what you see is drug particles and plasticizer dispersed in a wetting medium. In this case is polymer, and with the action of shear rate, and that too at a very high level, we need to disperse it uniformly in the matrix of polymer. That is what second picture on the right hand side indicates. So in a very simplified way, wetting means bringing two material close enough to adhere together. That is what we do. Either it will result in discrete particle dispersion or a complete solution at molecular level. However, to carry out this work, we need plastic behavior or viscous behavior of the mass, which is typically carried out by adding some amount of plasticizer. We have to select polymer which has plastic or thermoplastic nature. It melts, but it does not degrade. That is the tricky thing in the entire process. Next slide, please. What we see here is hot melt extrusion process is an enabling technology because it can help control dissolution characteristics. So we can either think of keeping crystalline drug substance as micronized particle, or we can even think of making a solution. And why we make this change in physical chemistry is to improve solubility behavior of the drug significantly. And typically, amorphous drug dispersed in water soluble polymer shows a very high intrinsic dissolution rate. In case of the transdermal preparation, it is used to enhance permeability. We all know the drugs are dispersed in permeability and control flux ratio also. Being a continuous process, I would like audience to give some importance about how it works. It is different from any batch process, and therefore endpoint is not critical here. What is critical here is steady state. Steady state, unlike endpoint, is a dynamic balance of many variables working together. So in the process, when we set up and when we make it stable, we have to balance so many variables. So it is always a multivariate balancing act which we need to do. Sometimes an operator or a manager finds it very difficult to do it. But based on our experience, once we give careful consideration of those factors, we understand what makes high impacting or moderate impacting or low impacting, and depending on those impacts, we try to scale those variables in such a way that the process remains stable during the entire run. Next slide, please. So these are some of the lab, pilot and commercial scale equipment, how it looks. If you see the quadrant on the top left, it's a tabletop hot melt extruder. It works with 50-gram material in an hour. So it is loved by academician researchers throughout the world, where you can keep it on your table and you can operate it with very small quantity and typically used in NCE product development. On the other quadrant on the left below is a lab scale equipment which is not tabletop, which is required to be installed. It also comes as a GMP compliant, and it has a typical throughput rate of about a kg per hour. Typical batch size in our lab is half a kg to 1 kg. On the right hand side top, you will see a pilot scale equipment, and that is used in technology transfer from lab to commercial scale. When we go, remember, it follows non-linearity. It is not so simple to scale up because there are so many variables which follows non-linearity. So we also provide a pilot scale equipment followed by a typical commercial scale equipment, which is omega 30 on the right hand bottom, is used typically with a throughput rate of 30 to 50 kg per hour, which is a decent throughput for manufacturing of any solid oral product. Next slide, please. So today, if you see, technology is at a crossroad. I started saying that although it was developed during Second World War, it did not get the recognition what it deserves. And if you see some of the reasons why it is at crossroad, is it has multiple limitations. For example, when we talk about shear, it has certain peak and trough. And until unless we have very good control built in the process through the selection of appropriate screw, we encounter that we are not able to run it at higher speed, which is a must for very good homogenization and mixing. And if we are not able to operate it at very high speed, then what happens? Kneading operation gets affected, and kneading cycle also gets affected, leading to longer residence time. Remember, you are running this process at a very high temperature, and if you end up with a longer residence time of few minutes, you carry a risk of burning drug or even polymer during the process, leading to undesirable output. So process capability in a continuous process is measured typically in seconds or minute. Unlike batch processing, where typically it is measured in amount or quantity of the material which it can handle. In a continuous process, we measure efficiency in terms of seconds or minute. Typically, till date, we have seen hot melt extrusion process with mean residence time of 1 or 2 minutes. We are able to improve it to a few seconds of mean residence time, and that I will talk in the next page. Please move to next page, please. So what is 21st century concept? 21st century concept is using the extruder as a mixing vessel, and that vessel is either for a dry grinder or a dough kneader or a fruit juice mixer, which is a less viscous liquid. We are using in our lab every day for dry grinding or even semi-solid mixing, highly viscous or less viscous. So we know how to use it. It comes with a very good understanding of all the elements of processor. We typically operate at a very high rate of screw speed. It means we are able to rotate it at a very high shear rates, and therefore we are able to take advantage of kneading cycles and stirring rate. All this comes with a proper understanding of various screw geometry. And when we talk about speed button, typically you see in any kitchen, mixer, grinder, it has many speed button. So what can happen at a very high speed? Typically we see that if we go down in speed, that type of activity or desirable event is absent at a low speed. So optimal mixing, many times we have seen, is obtained only at high stirring rate or greater kneading cycle. To get up to that speed, again, a lot of science behind geometry of the element comes, and therefore, for a pharmacist, it looks a more of engineering science than pharmaceutical application. Today, when we talk about 21st century processes, as I said, it runs with the mean residence time of 10 or maximum 20 seconds. And therefore, we are able to run it for those products which otherwise is very difficult to process because of degradation while melting. And therefore, we are talking about a situation where you think about frying ice cream. And we have seen that we are able to melt a product without much degradation, which otherwise is considered highly challenging. So in terms of improvement, what we did in last 2 decades in our lab is we have minimized the shear peak variation, and therefore we are able to run it at a very high speeds of operation, resulting in higher efficiency, ability to gain control of shear both in terms of quantitative and qualitative way, and we are able to increase kneading cycle, we are able to increase stirring rate, and therefore we are able to handle highly viscous or highly sensitive substance, keeping short residence time requirement. Therefore, if you see in recent uh, last 5 years, people are now using this processor much more, I would say, efficiently in many substances. And today's the efficiency of the processor has gone up one order of magnitude, which is 10 times. So what was not possible about 15 or 20 years back, today, you see those things are possible thanks to greater understanding of engineering sciences involved in application. And therefore, you will see that in recent time, there are many products where even polymer, which was considered very sensitive, now is being handled efficiently. For example, HPMCAS, which is hydroxypropyl methyl cellulose acetate succinate, which is considered very difficult to handle, that becomes easy to handle now, and drug development becomes unrestrained. That's our some of the improvement what we have seen in last decade. I will end up with a big thank you once again to APJ Satya University. I have tried to give a glimpse and of hot melt extrusion process. Next slide, please. However, I would like to keep it open to student to ask any question, because this is not a new process. This is something which has evolved a lot in last 20 years, and therefore my humble submission to a student is, please handle it, please use it as much as possible. And if you have any question in this area, particularly, Steer Life is always there to help you out with sound scientific understanding. Thank you so much for giving us time.
Thank you so much, sir. As we are running short of time, so may I request Navita ma'am, may I request Navita ma'am to please briefly summarize this session.
Sorry. Hi. Rani, can you just repeat that?
Yes, ma'am.
Yeah, Rani, can you hear me?
Yes, ma'am, we can hear you.
Yeah, can you just repeat? You asked me to?
Yes, ma'am. So I request you to please briefly summarize the session.
Sure, sure. So yeah, I would like to thank Mr. Indu Bhushan for a very interesting topic and presentation. So melt extrusion processes, like he explained, they've been used in industrial applications in several years, starting from polymer and plastic industry. Hot melt extrusion, has found numerous applications in pharmaceutical manufacturing practice. The development of HME process and applications, as we've seen, listening to the lecture, needs very sound understanding of diversified areas such as polymer science, rheology, formulation and development, process engineering and design, etc. So the most common application of HME processes in oral solids product development and manufacturing involve fusion and structural modification of the drug substance and functional ingredients used in the composition to improve or modify performance of the drug products in terms of its in vitro release, bioavailability, shape and size, taste, and stability. So today our speaker has explained to us the fundamental principles and critical factors to be considered while developing melt extrusion processes using co-rotating twin screw technology, various types of mixing principles and actions typically observed while using the processor to provide both thermal and mechanical energy to bring about material transformation dependent on physico-chemical nature of the ingredients have also been presented. Additionally, Mr. Bhushan also gave us an introduction of the process design and development in technology to address multiple limitations based on his own experience for a wider application and an improved efficiency. So thank you so much. Any questions?
Thank you so much, and thank you so much, sir. Now, we from University, we are felicitating our resource person, Mr. Indu Bhushan, sir, with the certificate of excellence. Please accept it, sir.
Thank you.
Thank you, sir. And now we are moving to our next speaker, Dr. Kanchan Kohli, ma'am. Dr. Kanchan Kohli, ma'am, is the Director of Research and Publication, Faculty of Pharmacy, Lloyd Institute of Management and Technology, Greater Noida. So may I request, ma'am, to please share your PPT and start your presentation.
So, good afternoon or good evening, so. Am I audible?
Yes, ma'am. Good evening, ma'am.
Okay. Good evening. First of all, I'd like to thank organizers of the program, that's the DST-SERB, and chairperson, co-chairperson, Ms. Navita and Dr. Majumdar for chairing the session. And as the well conversed since morning, you've been listening about the nano one, whether the medical devices are there or the medicines are there. So at the end, I'm also winding up with the nanomedicines also. And that's the nanomedicine and future perspective. So this is just the pick which I have picked up for various nanoparticles. So what's the main object of this our presentation? That's the introduction to nanomedicine, history and timeline, application of nanomedicine, advantages and disadvantages, challenges and conclusion. And by this you must by this time all are very aware of what's the nanomedicines and how what are the various products which are available in the market till now, and for what are the various products which are under pipeline also, for which the preclinical studies been conducted, and after preclinical, maybe they are in the clinical trials itself.
and they are yet to be launched in the market. So they are the nanomedicines. Generally, we are, having this nanotechnology, biology, chemistry. In these, the nanomedicines application is there, or in which, through which we can have the nanomedicines. So human body is basically an extremely complex system of interacting molecules. There's the molecular machine. Then therefore, technology requires to truly understood and repair the body is the molecular machine technology. That is the nanotechnology. So nanoscales for which we are using it. That's the nanoscience, which involves research and technology development at 1 nanometer to 100 nanometer range. So nano indicates 10⁻⁹. In nanometer is 10⁻⁹ meter. 1 nanometer is a billionth of a meter or a millionth of a millimeter. A single human hair is around 80,000 nanometers thick. A typical protein size lies between 3 to 10 nanometers. Red blood cells are a standard size of about 6,000 to 8,000 nanometer. That means it is existing, already was existing, but we had now come to know that's what the nanomedicines could be also there, like this way. So nanomedicines, the official definition of the US National Technology Initiative says, nanotechnology involves research and technology development at the atomic, molecular or macromolecular levels in the length scale of approximately 1 to 100 nanometer range. European Science Foundation has defined nanomedicine as the science and technology of diagnosing, treating and preventing disease and traumatic injuries, of relieving pain and improving human health using molecular tools and molecular knowledge of human body. Aim of nanomedicine: comprehensive monitoring, control, construction, repair, defense, and improvement of all human biological system working from the molecular level. So using engineered devices and nanostructures ultimately to achieve medical benefit. So history. When we are going for the nanomedicines or the nanotechnology itself, so there is little bit history behind it. There's on December 29, 1959, our physicist, actually it is started from the physics only. Physicist Richard Feynman gave a radical structure at an American Physical Society meeting at cultural title, "There is plenty of room at the bottom." Feynman suggested that it should be possible to make machines at a nanoscale that arrange the atoms the way we want and do chemical synthesis by mechanical manipulation. This lecture was the birth of idea and study of nanotechnology. Continuation with this, then Professor Norio Taniguchi of the Tokyo Science University introduced the term nanotechnology in 1974. He described nanotechnology as the processing of separation, consolidation, and deformation of materials by one atom or by one molecule. Again, in the 80s, Dr. K. Eric Delexer promoted nanoscale phenomena through books, Engines of Creation, The Coming Era of Nanotechnology, Nanosystems, Molecular Machinery, Manufacturing, and Computation. He was ultimately responsible for the term nanotechnology to acquire its current sense. He was presented the first PhD in nanotechnology. Again, Hollywood provided the public with a glimpse of the future of nanoscience with the release of the film Fantastic Vogue in 1966, which depreciated a surgical team that was miniaturized and injected into a man to operate a blood clot in his brain. So, as we go for the Ayurveda, we did have Bhasma from the ancient time. So now when we quantify the bhasma, they could be the nano sizing, or they could be the nano sizing, but there is the mixture of the nano or micro or more could be there, but ultimately they can be screened out and they can be further converted to the nano sizings. So bhasma is a Ayurvedic metallic mineral preparation treated with herbal juices or decoction and exposed for certain quantum of heat as per Kuta system of Ayurveda. Bhasmas are claimed to be biological produced nanoparticles, and this comes now the ethno-nano medicine. So nanotechnology timeline, these are the two, we can say, the journals which are now, the leading journals. There's the Nanomedicines or the books, European Technology Platform. So these are the leading journals from the nanotechnology only. So nanotechnology timeline, the US National Institute of Health, NIH, released their first roadmap on nanomedicine in 2004. In 2004, the National Cancer Institute, as part of NIH, launched the Cancer Nanotechnology Plan, a strategic initiative to transform clinical oncology and basic research through the directed application of nanotechnology. So this is the first product that was produced by NIH only, and that's the Abraxane, which is the albumin-bound paclitaxel. There's a taxane, and this was the first product through Nano, which has come out in the year, I suppose, 2005, and it was approved in 2005 by FDA, and then which was launched into the market, and that was for the breast cancer purposely, but it was in the nanoparticles, but still unable to answer the mechanism of the how it is lowering the P-glycoprotein protein levels. This is still it is unknown, and still kind of the searches are going on. Current status, the market analysis reported that market for the nanomedicine is continuously growing at the rate of 28% with a 35% increase in the revenues generated. The National Science Foundation estimated the market for all nanotechnologies to be $1 trillion. Huge investments are being made in nanomedicines to develop novel therapeutic deliveries. Currently, more than 38 nanomedicine-based products are on the market, which we'll see later, with estimated sales of $6.8 billion, and the efforts to bring them on the market are increasing continuously. As I mentioned, many products are still in the pipeline, which has to yet to become into the market. Then we are having these many products, and the name of the companies are there. I'm not repeating, just you can go through it this, and they are the nanotechnology products, and mostly the projects, they are in the liposomes form, which the product paclitaxel, which I told you, that is the albumin bound nanoparticles, and we did have the nanocrystals based on the nanocrystal technology also, which in the morning you had a very good session also, and kind of we are having anti-cancer products, antifungal products, then mostly they are the anti-cancer or the any neoplastic products so far, which are available in the market. So application of nanomedicines, they could be used for diagnostic purpose, imaging and identification, therapeutic, delivering medication to the exact location, killing of bacteria, viruses and cancer cells, repair of damaged cells, and we are having the theranostics agents also, which we'll talk later on. So these are the some of the examples or the applications in vitro, in vivo, as a diagnostic agents, as a therapeutic agents, as tissue regenerations, products which are, still the many we had a session, you had a session from nanofibers also, and many more preparations will be coming soon to the market. So diagnostic use, imaging, nanomaterials are being used extensively as contrast agent in non-invasive medical imaging tools, including computed tomography, magnetic resonance, posterior emission tomography, single photon emission computed tomography, ultrasound, and optical imaging. The contrast agent used nano-sized metal oxide, dendrimers, quantum dots, etc. So diagnostic use, again, continuation with that, quantum dots, nanocrystals, they are the nanocadmium and nanozincs, and they are semiconductors used to tag biological molecules, have application in medical diagnostics, targeting therapeutics, and high throughput drug screening. Tiny crystals produce sequences of colors when subjected to ultraviolet rays, as you are seeing in the pics. Currently, in vitro diagnostics are costly, and it is hoped that the new generation nanoscales lab-on-a chip will offer advantages, including reduced cost, portability, and shorter and faster analysis. Applications may include measurements of saliva for periodontitis, heart disease, insulin detection, and improving healthcare accessibility. As very soon we are coming to know that even corona that can be detected through saliva. I mean, some researches are going on. Diagnostic, again, a lab on a chip with the size of a postage stamp is already commercially available and is used to monitor lithium medication levels for manic depressive patients at home at much lower cost and with greater convenience. At home, now you were finding the kits are available at home by which the detection can be done for many disease. Other diagnostic application using nanotechnology and already on the market include colloidal gold particles, which due to their stability have been widely used to rapidly test for pregnancy, ovulation, HIV, and other indications. Magnetic nanoparticles used for cell sorting application in clinical diagnostics. Magnetic iron oxides nanoparticles for magnetic resonance imaging, first approved in Europe in 1993. Then magnetic iron oxide nanoparticles also show great promise in the detection of Alzheimer's plaques. Then in medical instruments, as since morning, medical devices, nanomedical devices is also one of your lecture. So medical instruments are also being increasingly miniaturized with nanomaterials for increased efficiency. Carbon nanotubes may be used instead of glass pipettes for delivery into cells. Nanosilver is increasingly incorporated into catheters and other instruments as a coater because of its strong antimicrobial effect. Even maybe in coming time, masks also, face masks, the coating could be done with the nanosilvers. Wound dressings may also incorporate some part of nanosilver for the same purpose. Therapeutic uses, drug delivery using nanoparticles, that's nanopharmacology, and application of nanotechnology to the development and discovery of drug delivery methods, applications to deliver medication to the exact location, lesser side effects, and this we all know why we are going for the nanomedicines, lesser side effects and high efficacy, improves bioavailability, molecular targeting by nano-engineered devices. Nano formulations of existing drug can overcome common pharmaceutical problems by increasing solubility, limiting systemic toxicity, increasing bioavailability, and improving immunocompatibility and cellular uptake. Nanotechnology has been also used for targeting drug delivery, which will allow controlled drug release at just the right place and dose, improving patient safety and compliance, and reducing side effects. Drug delivery vehicles under investigations includes polymeric particles, dendrimers, nanoshells, liposomes, micelles, and magnetic nanoparticles. And out of this, which I find the liposomes are commonly used as a nanomedicines for many of our drugs. So advantages of a targeting drug delivery system: encapsulation of a drug such as a liposomes or micelles can avoid local irritation and decrease local and systemic toxicity. What the disadvantages? The drug delivery mechanisms may also introduce toxicity of their own and have unintended side effects. So metal-based nanoparticles, we are having gold, silver, cadmium, zinc. Lipid-based nanoparticles, liposomes and neosomes-based. Polymer-based nanoparticles, dendrimers, chitosans, micelle-based. Biological nanoparticles, we are having arginine glycosporic spartic acid, RGT peptides-based. They are mainly peptides-based. So liposomes, they are the small spherical vesicles composed of lipid bilayer surrounding aqueous inner face. So they are normally consist of the phospholipids and cholesterols, and size is 200 nanometers or smaller. And applications targeting drug delivery, cancer treatment, and some liposome-based pharmaceuticals, amphotericin B, which was widely used during fungal infection during COVID time, and still it's being used for fungal infection, dinorubicin, doxorubicin, and amikacin. So from this example, you can see we did have two amphotericin preparations, simple one and the liposomes one. But every time the demand was for liposome preparation. That was the reason, what's the shortage of the liposomic preparation in the market was there, whereas the other preparation was available. But even the physicians, patients, they were all well aware of the amphotericin liposomic preparation. So because of its targeted effect also, and the quick effects also, and the dose was very small as compared to the simple formulation. So next we are having the pegylated liposomes, and they are also being commonly used nowadays, makes the liposomes less vulnerable to immune system. And pegylation, by increasing the molecular weight of a molecule, can impart several significant pharmacologic. What are the advantages over it? Improved drug solubility, enhanced protection from proteolytic degradation, increased drug stability, extending circulating life, reduced doses frequency without diminishing efficacy with potentially reduced toxicity. Next, please. So niosomes, they are non-ionic surfactant vesicles, widely studied as an alternative to liposomes, similar to liposomes in terms of their physical properties, but they are being little more stable as compared to the liposomes. They have the potential for controlled and targeted drug delivery. Niosomes enhance the penetration of drug. So little bit advantages is there through over the liposomes. So we are having the dendrimers. Dendrimers are again man-made molecules having a tree-like structures. So here we are having dendrimers, they are measured 2 to 20 nanometers. They are the nano devices, but still we didn't have any marketed formulation. Only except one formulation is there, that is the antifungal preparation. Otherwise, US FDA has not approved dendrimers yet. They are just in the pipeline. So nanoshells, again, we are having the core of silica and a metallic outer layer. Still, they've been not approved, and again they are under only under the, we can say, you are the research based only. So carbon nanotubes, again, in the carbon nanotubes, they are the kind of devices such as sensors and provide a novel means of delivering drugs with great specificity. But when we coming to the toxicity of these nanotubes, carbon nanotubes, that is much, and scientists are searching the way that how we can further reduce the toxicity of nano tubes. So these are the nano drugs, dendrimers, which are under pipeline, under the maybe preclinical studies stage, and these are the liposomic preparation which are under pipeline also, and which been available in the market also through some companies under pipeline, and others been launched into the market. And nano drugs, solid lipid nanoparticle, still we don't have many preparation out of it except one of the dermal preparation, and again they are under preclinical stage. And micelles preparation also, we don't have much preparation in the market. Again, they are into the pipelines only. So polymeric nanoparticles, celecoxib, then these preparations, again, they are under the pipeline, not many preparations, but only the paclitaxel albumin particles which are available in the market. Then we are having the nanocrystals. These are the preparations which are available in the market. Then we are having the FDA approved nano drugs. These are the drugs which are approved and which are available. Liposomes, we have already discussed, from amphotericin B, doxorubicin, doxorubicin, citrabine, morphine, ampicillin. From emulsion, we are having the cyclosporine A. Engineered nanoparticles, we are having the diclofenac. Polymeric nanoparticles, we are having the paclitaxel. Nanocrystals, rampamycin, araptine, fenofibrate, magastrol. These are the drugs which are available. Drug discovery, nano and micro technologies are part of the latest advanced solution for decreasing the discovery and development times for new drugs and potentially reducing the development cost. Next slide, please. Therapeutic uses, then this can be used for the cancer treatment, which we have already discussed it. And we can use for the localized also, and for the systemic treatment also. And thermotherapy, a form of cancer treatment that uses heat to destroy cancer cells. Then we are having the tissue repair and replacement. They are the implant coating, biocompatible nanomaterials and coating to increase the adhesion, durability, and lifespan of implants. Nowadays, even nanoparticles are only available. So then we are having, next please. They can be used nano robust, future vision. There's a nano robots are nano devices. They can have the potential application include to repair or detect targeted damages and infections, early diagnosis and targeting. These are the some, the hypothetically also been made, and a microscopic machine roaming through the bloodstream, injecting or taking samples of identification and determining the concentration of different compounds. Again, the robotics are there. Robotics is there, which inspired into the lungs and attached to the tissue surface, and that could be inhaled. Mechanical drilling of a small tumor mass by a nanorobot. So these are the kind of the coming field into the nano one only. The nanorobot, advantages of nanomedicines. Of course, now we know that all the advantages: drug delivery, exact location, lesser side effect, high efficacy, molecular targeting, detection, no surgery required, disease can be easily cured. Disadvantages: not practical yet, high cost, implementation difficulties, nanotoxicity. Then nanotoxicity is there. These are the where the nanotoxicity can be there. So these are the areas where the toxicity could be there. Then again, we can have the disadvantages of this. Then challenges. What are the challenges? Regulatory issue, environmental issue, social issue, ethical issues. So from regulatory issues, we are having the, from FDA side, what the issues could be there, they are highlighted, but not much time to discuss it. So I'm just skipping up some of the slides quickly. Environmental risks are there because it is in the nanoparticles. So the flowing and the dispersing is very easily, and we are having the ethical issues. There's the bioethical research believe that nanomedicine could be manipulated to harm the human body rather than healing it. So these things yet to prove it. So conclusion, although realization of the full potential of nanomedicines may be years or decades away, recent advances in nanotechnology related drug delivery, diagnosis and drug development are beginning of change the landscape medicine. The possibility are endless, but will take time to develop. Nanotherapies could in the long term be much more economical, effective and safe, and could greatly reduce the cost of current medical procedures. So nanomedicine is the future medicine. So again, I mean, the concluding, human health has always been, that's the Richard S. Smiley, human health has all been determined on the nanometer scale. This is where the structure and properties of the machines of life work in every one of the cells, in every living thing. The practical impact of nanoscience on human health will be huge. So thank you for patience listening and giving me an opportunity to share my views with you people. So thank you again for patience listening. Any question would be there that can be answered.
Thank you very much, ma'am. Can we ask Navita ma'am to kindly
That very quickly, I have just
summarize the topic or share a view on this topic? Navita, over to you.
Sure. Thanks, Dr. Devan. Thank you, Professor Kohli. That was a very comprehensive and interesting presentation of nanomedicine, and I should say a very appropriate topic to be the last session of the day. So our speaker has shared with us the origin and evolution of nanotechnology, specifically nanomedicine, its purpose and also its advantages and disadvantages. She has also educated us on where we stand today in terms of current status, as well as, the examples of various formulations which are available and those that are in the pipeline. She has also shared with us the potential applications in the areas of clinical medicine, drug discovery, drug delivery, therapeutics, diagnostics, and also in tissue regeneration. So while elaborating about these future opportunities, she has also shed light on the regulatory and ethical challenges around its use. So, I found it to be a very interesting session, and thank you so much once again.
Welcome.
Any questions from the audience? There are no questions since they are just everyone getting ready to catch the bus.
Yes.
The time, and really want to thank Ma'am Kanchan for accepting our request to be present as a resource person today. Thank you very much, ma'am. It's actually from an academician to the student, the basic things which are required is very much summarized. Just now, one of the faculty also suggested that, ma'am, if you have the slides, we can share and one day run to our third year and fourth year students so that they can gain more knowledge. We will be doing because we have your slides. Thank you very much.
Welcome.
And
welcome.
Thank you very much. Navita.
Thanks all.
Really kind of you and for accepting our request and moderating the session. Thank you very much.
My pleasure.
Thank you so
much. We are moving on to the next. Thank you very much. And we had poster evaluation today. Dr. Gaurav Jain and Dr. Aparna has evaluated. We had received around 50 posters through online and offline mode, and 200 participants have participated from various universities like GD Goenka, Jamia Millia, DST-SERB, MVN, PDM, KR Mangalam, ICMR Patna, Mewar University, IFTM, Advanced Institute of Pharmacy, Seth Vishambarnath Institute of Pharmacy, Lucknow, Chitkara University, and GNU Delhi. My sincere thanks to all of our participants who have come from such esteemed institutions. Now coming to hold your breath, the result of the poster presentations. Now, the first group, which includes our senior M.Pharm, PhD, and PhD, in that the first prize goes to Ikra J. In the treatment of melanoma. May I ask Dr. Majumdar to kindly come on the dais and Ikra also to please present. The certificate will be emailed to you shortly. Now, the second, Ikra is from which? Jamia. Wonderful. The second prize goes to Kavish. Kavish, who has spoken on formulation, optimization, and evaluation of borage oil-based nano-emulsion dosage formulation. Can you ask Kavish to come on the dais? Or someone to receive on behalf of Kavish? Kavish from which university? Jamia. So can I ask Ikra only to receive Kavish? Yeah, thank you. The third, Ruchika Kalra. Is Ruchika available? So she is from Advanced. She has spoken on, they are presented on that potent anti-dandruff essential oil, the nano-based formulations. Now coming over to group 2, the first prize in group 2 goes to Sneha Anand. Is she there? Student, okay. Then second to Prem. Saint Visambardhar University, Lucknow. Yeah, please come on the dais. Actually, I student have the application, so I have to ask you. So now the third prize goes to Manpreet Singh Bedi. Is he our student? ASU? Yeah, please come on the dais. Thank you very much. So we are coming almost to the close of the day, but the day will not get closed unless until we have really our scientific abstract book for which the abstracts have been evaluated. Now I request Dr. Majumdar to stay on the dais and Gigani, please come on the dais to release the abstract book. Can I have the abstract book? And let the show begin. For the release of the book, I am joining you. With me, the book has been created by the scientific committee. So both the chairperson and co-chairperson of the scientific committee, Narendra and Gupta, please join. Thank you very much. We enjoyed release of the abstract book. And now to continue, I have see one thing was said. वो कहते हैं ना कि रोते रोते आए थे सब हंसते हंसते जाना है जो हंसते हंसते जाएगा वो मुकद्दर का सिकंदर कहलाएगा। सम सॉन्ग वाज देयर, है ना? तो ऐसे ही अनफॉर्चुनेटली हम आए थे और फॉर्चुनेटली एक अच्छा सा इवेंट कंक्लूड करके वी आर लीविंग फॉर द गाइस, एंड फॉर दैट मैटर the thanks are to my team. And if I do not thank them, my day will not get over. So foremost, I will ask Dr. Majumdar to come on the dais. हम दोनों का काम है, हमें तो mentorship करनी है. Please come on the dais. And for the senior most faculty member, and she has been handling the technical program committee, Dr. Moini, please come on the dais. May I ask Dr. Majumdar to honor her with a certificate and a memento. Thank you, Dr. Majumdar. The event organizer of SPS, the charming lady, please, Dr. Rani, please come on the stage next. A certificate of excellence and a memento to go over there. The technical man, scientific committee head, Dr. Narendra Yadav, please come on the stage. The man who is so busy, always working on something another, and the students are calling him Guruji. Please, Guruji, come on the stage. Manoj, Narendra is almost can't work without Gufran. So Dr. Gufran, please come on the stage. अब गुफरान की गाड़ी चले ना हसन के बिगाड़. हसन कहाँ है भाई? Where is he? He's again गायब. See, your fellow is not there, but in an absence, we always discuss that. Okay, and Lupa, actually she met with the COVID. She has really got infected, and a day before she was positive. In her absence, it is being given to her. Now, coming over to the next. The co-event manager with Dr. Rani, Mukesh, please come on the stage. Now next is the one who has so well arranged all the screens here so that the online and offline goes very well. The new member in our family, but has been adjusted in 4 days so well that it seems like he was with us from ages. Sumit, please come on the dais. Another new member in the family joined Rashid, but has been very busy since morning for the registration committee. I think is Rashid. If you can hear me and come on the dais. Even now he is working with your feedback form and certificates. So we will be handing over to him shortly. Rashid, if you are there, please come on the dais. Okay, thank you very much. Moini, you want me to do something else? Anything else we have to do? No, then come and please thank all of us, at least to me also. Please come, and then we will be after that standing for the national anthem, and meanwhile the technical committee arrange that, and definitely we will take another few minutes. Thank you very much. Definitely our anchors of the day, 4 of them, they have very done wonders. Can I ask them to come one by one? That is what I was saying. I was missing on something. So please come, please. Dr. Majumdar, to please encourage them. Niharika, please come on the stage. Aditya, Rashmita, and Shania. They've really done good job today. And this is not the end, this is just the beginning. And they are in first year, I think. 2 of them are in first year. Ah, wonderful. That's wonderful. A round of applause for 4 of them. They were the hosts. And Mohini, she is really helping you out. Please tell her to come on the stage. Mohini, unko bhi do Mohini. The one who has been giving to all of us. Please come. Monica, I am right. Monica, please come on the stage. Thank you. See, we were holding the mic for Rashid and Rashid has actually come. So now, Rashid, please come on the dais. Now, with Dr. Majumdar, he is about to give you something. A certificate of honor and the moment to you. एक तो ज्वाइन कर दी, मिल गया बॉस, बाकियों को तो बहुत टाइम लगा. Thank you very much. Thank you, wonderful.
Good evening, everybody. On behalf of School of Pharmaceutical Sciences, I would like to extend vote of thanks. First of all, my sincere thanks to Almighty God for making today's event a grand success. By His blessings and grace, we are able to make this event what it was. I would extend my thanks to respected Chancellor Ma'am, Mrs. Sushma Paul, for giving us the opportunity and platform to organize one-day national seminar. I also wish to express my gratitude to VC Sir, Professor R.S. Dhankar, Dean Ma'am, Professor Anupama Diwan, and Emeritus Professor Dr. D.K. Majumdar Sir for initiating the event and providing encouragement at each and every step. We would like to thank our distinguished keynote speakers, Professor Alok R.A., Dr. Kalaya Selvan, Dr. Manish Diwan, Dr. Arvind Sir, Dr. Dheeraj Kumar Chopra, Professor Bansi V. Malhotra, Dr. Indu Bhushan, and Professor Kanchan Kohli, who have graced the occasion by sharing their knowledge in simple and effective way, so that we all were able to understand and the concepts easily. We extend our thanks to Session Chair Dr. Mukhe Khar, Professor Harvinder Putli, Dr. Jitendra Madan, Professor B.K. Majumdar. They have summarized the concepts very effectively. A great and special thanks to Professor Sanjay Agarwal, Dean SJMC, and their team for audiovisual coverage of the event. A great thanks to organizing committee for putting together the wonderful event to make us understand the intricacies of complex subject nanomedicine. A sincere thanks to DST and SERB for sponsoring the event. Thanks to Walter Bushner, Martin and Harris, Global, and ASG Biochem for sponsoring the event. A special thanks to faculty members from other colleges who have joined us and participated. Thanks to all the budding pharmacists for being patient listeners and absorbing knowledge disseminated. On behalf of School of Pharmaceutical Sciences, APJ Satya University, I would like to thank you all for joining the event and making it a success. Next year, we'll again meet for the 4th event in the series. This is only a curtain raise. Thank you all and have a nice week. Thanks.